All right, looks like we are live. And I want to welcome everybody to Standing for Truth. My name is Donny, and this is Sam. We are your hosts and moderators for tonight's open mic debate challenge on one of our favorite topics, genetic entropy. It is a privilege to have Paul Price here for this much anticipated event. We've had this set on the channel now for somewhere between a month or two. Genetic entropy is currently undefeated. And so this is an opportunity for all skeptics and critics to join and to bring your best objections, arguments, and criticisms for Paul and at times Sam and myself to engage. Paul, you've written on this topic. You've also debated on this topic going back a couple of years now. Time flies. You had a formal debate with Dr. Ron Garrett on genetic entropy, and you've written numerous articles on this topic. So it's great to have you here for this open mic event. Paul, how you doing tonight, my brother?
I'm doing great. I'm excited to be part of this. I'm always excited to get to be taking part in your amazing channel, Donny. It's what you're doing here, I think, is it's definitely unique on YouTube. You know, there's not many other channels that you could lump together with, I think, with what you're doing here. So, you know, that's why I think you're continuing to grow and be blessed with more and more subscribers. And I hope to see that continue because what you're doing is worth doing. That's the bottom line. It's a great. You're doing work for not only the church, but for those outside the church who are looking for answers, who want to be able to believe in God, but there's something that is standing in their way. There's something that is giving them trouble and doubts and stumbling blocks, and we're here to take those away.
Amen. Paul, I appreciate those kind words. All the glory to God. It really is a blessing to have fellow brothers in the Lord like you willing to join us here in what I like to call the debate dojo in these live shows and really help edify the body of Christ and also bring those who are on the fence, those that are agnostic to the truth. Sam, my co-host for tonight, how are you doing, brother?
I'm doing good. Thanks for having me. It's a pleasure to be here with you guys. And yeah, I just hope that this stream gets people thinking, especially the critics, about being reconciled to the God they know.
Amen. Well said. Paul, you're definitely no stranger to this channel at this point. I've got a whole playlist with all of your previous shows, your formal debates, your open mics that you've done with us, your presentations, and you'll be back here next month as well. So this is something a little different, though. Usually we're engaging science-related topics. So this will be more into the theology world. Are Christians under the law? Sean Griffin, popular YouTuber, and yourself. I think this is going to be a real fantastic.
That one might turn out to be that might have come out sounding arrogant, so I'm not going to say it. But I am looking forward to the debate. How about that?
It may just be the debate of all debates. We'll put it that way.
And also I am humbled that the great Pastor Marty is here with us tonight in the chat. I just saw that. So thank you.
Yes,
thank you, Marty, for being here.
And I agree. Paul is a well-dressed man.
Yes.
You always come equipped for these discussions. Paul, again, to the audience, do check the description box. I should have at least one or two of your articles linked, Paul, there on the topic of genetic entropy. And so I would say you are in many ways an authority on this topic as you've been studying it for many years. You've been engaging the critics as well for many years. So I think this is going to be a lot of fun. I will go over the format for the audience. And so we are going to be starting off the show with an important presentation, about 45 minutes from Paul on the topic of genetic entropy. Then what we'll do is open up the mic. And so sometime between now and maybe 30 minutes from now, I'll post the StreamYard join link into the chat. And so for those interested in joining, make sure to click the link. I'll have one at a time engaging Paul, and then I'll have a second evolutionist on deck, basically, who will be to the right of our StreamYard template. Sam and I will be hosting and co-modding, but it's not a formal debate. So if we feel compelled by the Holy Spirit to make a point or add something, then we will certainly do so. And so, okay, with that, Paul Price, if you had any other words of introduction, feel free to provide those. And then I think we'll just get right into your presentation.
No, let's just get right in because there's a lot to cover here. And Donny, I always like to ask, am I coming up here? It looks like the share is working here. So
it's coming in great, Paul.
All right, very good. Let's get started. Okay, tonight, this is the mother of all topics when it comes to creation apologetics, in my humble opinion. And that is because what we're doing here is that we are getting up under the hood of neo-Darwinian evolution, the neo-Darwinian synthesis, as they call it. This is the mystery of the genome. How do we explain how evolution is supposed to work on a base level? And I also need to address this question because I'm sure going to have to, be criticized that I, being a non-specialist, am speaking on the topic of population genetics. And, there's no need to hide or be concerned about the fact, yes, I am an apologetics generalist. I am not a scientist. I do not possess an advanced degree that is relevant to this topic tonight. But I think if you look at the history of science, you will find that there have been many, many, many times in history when people have made contributions or even founded entire fields. Not that I'm claiming to do any such thing, but it's well known that Charles Darwin himself did not possess, a degree relevant to the field that he was speaking in. And unfortunately, many people who are specialists fail to look at the big picture. So what I'm going to do here tonight is help everyone by way of Dr. John Sanford's work to look at the big picture of evolution and connect the dots. I'm going to be quoting from specialists in population genetics to show the validity of Dr. Sanford's argument. They do see the dots, and there's no question that they see the dots. But the one thing that they cannot do is, they will not do, rather, is to connect those dots. And it's also the case that it wouldn't matter if I did happen to possess a PhD in genetics. The haters, and there's always going to be haters, they would still refuse to listen. That is just the way it is. After all, they are refusing to listen to Dr. Sanford himself. So, if I were sitting here with an advanced degree, it would make no difference here. We don't need to use arguments from authority. We need to look at what, the consensus of the most, the most educated people in this very narrow specialized field of population genetics and mutation distribution. What do they say? Are they willing to admit to the dots, but they just won't connect the dots? Now, Dr. Sanford himself anticipated this situation. He wrote, "I realized that I had a moral obligation to openly challenge this most sacred of cows. But I also realized that I would earn for myself the intense disdain of most of my colleagues within academia, not to mention very intense opposition and anger from other high places. To the extent that the primary axiom can be shown to be false, it should have a major impact on your own life and on the world at large. For this reason, I have dared to write this humble little book, which some will receive as blasphemous treason and others as revelation." So what is the primary axiom? This is a term that Dr. Sanford has coined, and it just simply means life is life because random mutations at the molecular level are filtered through a reproductive sieve acting on the level of the whole organism. And Dr. Sanford developed what I believe is a tremendously helpful analogy of a red wagon on a robotic assembly line. And we have the red wagon here on the left, and on the right you see the instruction manual. Now this instruction manual codes for, tells us how to construct the red wagon, as well as all of the component parts of the red wagon. Now, as this red wagon is going to go down your assembly line, there are two robots, one at the end and one at the beginning of the line. Now, the robot on the end of the line, we're going to call the judge. And what it does is that it examines the final product, but it does not read the manual. And the manual, by the way, as you could see there, it's actually going down the line with the red wagon. It's attached to it. On the other end of this robotic assembly line, we have the scribe. And it doesn't really read the manual either, but what it does do is it copies the manual. Now, if there's an example of unworthy product, our judge is going to destroy that product. But if we have something that passes the test, then the judge will detach the manual and send it back up to the scribe. At that point, the scribe is going to copy the manual with mistakes and then destroy the original. This process is evolution. And as it iterates time and time again, we can imagine that more and more mistakes are going to occur in this manual that keeps getting transmitted back and forth. And we can imagine that over time, those mistakes might build up to very big changes. As you can see, we've gone from a little red wagon to a much bigger, more complex red wagon. But it won't stop there, because this process has no end. And eventually we can imagine that we'll get a space station out of this process. And keep in mind that the space station is really nothing more than a mutant little red wagon. Can you believe it? Do you really think, now that you have seen what evolution is on a fundamental level, do you think that's credible? And in my opinion, it's pretty obviously not credible, but we need to figure out where the problem is. What is it that is preventing this from being credible, from being plausible? So here are the different stages of Dr. Sanford's analogy. Let's look at each one of them. The scribe would represent DNA replication. And this is not controversial. Both creationists and evolutionists agree that DNA replication happens. So there's not a problem there. We also both agree that there is a genome. That's the manual. So the genome is the complete set of all your genetic information. And then the little red wagon would represent a comparatively simple life form, perhaps like a bacteria, compared to the space station that we saw come out at the end. That would be more like a human being. Now, what about the mistakes? Now, it's definitely the case that mutations happen. So creationists and evolutionists don't disagree that mutations happen. But we do know that there are good reasons to question that mutations have the power to do what evolution requires them to do. And what about the judge? Well, that would represent natural selection, which is really just differential reproduction. And there's also good reason to doubt that the judge has the power to do what evolution requires as well. So here are the dots that we're going to connect tonight. These are the basic premises of Dr. Sanford's argument. Number one, mutations are the source of the raw material needed to drive evolution. Number two, the vast majority of mutations are damaging or deleterious. And conversely, almost no mutations are beneficial. And lastly, there are no strictly neutral mutations. But there does exist a class of mutations called effectively neutral, aka nearly neutral, whose effects are too subtle to be seen by the process of natural selection and are therefore free to accumulate in populations over time. These these effectively neutral, comprise the majority of mutations, especially for higher forms of life, for multicellular and up from there type of organisms. So our first expert witness here tonight is going to be Dr. Carl Heilbronn, PhD, Evolutionary Genomics. And this is our first premise. Mutations are the ultimate source of genetic variation that natural selection acts upon. Understanding the rate at which mutations arise and the distribution of fitness effects of spontaneous mutations is therefore of central importance to the study of evolutionary biology. Okay, we're going to move to the second premise now because that's pretty uncontroversial. So the second premise is that most mutations are harmful. Now, Dr. Sanford gave us an example here of a type of set of instructions that you might see in a factory for building something. And these underlined points would be highly impactful areas where you could have an accidental copying error and change something very fundamental, very impactful here. Step number 6, when you have completed the last step, go back and repeat step 3 until part B is 10.004 millimeters thick. Then wait no less than 3 hours before going to the next step. Now, if you imagine random or undesigned, unplanned changes happening in these underlined areas, obviously, such undesigned changes are overwhelmingly more likely to harm the end product than they are to improve upon it. And other experts agree. Dr. Philip Garish, PhD biology, writes, even the simplest of living organisms are highly complex. Mutations, indiscriminate alterations of such complexity, are much more likely to be harmful than beneficial. Anthony Griffiths, Professor Emeritus of Botany, University of British Columbia, this is a textbook author. He writes, the constellation of possible ways in which point mutations could change a wild-type gene is very large and varies according to the particular structure and sequence of the gene. However, it is always true that mutations that reduce or eliminate gene function, loss of function mutations, are the most abundant class. The reason is simple. It is much easier to break a machine than to alter the way it works by randomly changing or removing one of its components. For the same reason, mutations that increase or alter the type of activity of the gene or where it is expressed, gain of function mutations, are much rarer. Dr. Peter Kitely, PhD genetics, and Dr. Michael Lynch, PhD ecology, write, in summary, the vast majority of mutations are deleterious. This is one of the most well-established principles of evolutionary genetics, supported by both molecular and quantitative genetic data. Now, Dr. Sanford himself, he is also an expert in this area, and he writes, during the last century, there was a great deal of effort invested in trying to use mutation to generate useful variation. This was especially true in my own area, plant breeding. Millions of plants were mutagenized and screened for possible improvements. Assuming the primary axiom, it would seem obvious that this would result in rapid evolution of our crops. For several decades, this was the main thrust of crop improvement research. Vast numbers of mutants were produced and screened, collectively representing many billions of mutation events. A huge number of small, sterile, sick, deformed, aberrant plants were produced. However, from all this effort, almost no meaningful crop improvement resulted. The effort was for the most part an enormous failure and was almost entirely abandoned. Dr. John Sanford, PhD, Plant Genetics. So let's move now to our third premise, which is discussing nearly neutral mutations. Why is it the case that most mutations cannot be selected for or against by natural selection? Dr. Sanford has once again given us a great analogy, and this is called the princess and the nucleotide paradox. Now, there is an old fairy tale. I don't know how many people are still aware of it, but in the fairy tale of the princess and the pea, a prince wants to test a lady who claimed to be a princess to see if she was a real princess or not. He placed a single pea under a whole stack of mattresses on which She slept. The next day, she told him that she had a sleepless night because of something hard in the bed. He concluded that only a real princess would be so delicate as to be disturbed by that one single pea underneath a whole stack of mattresses. And this is analogous to the situation that we are in with natural selection and single mutations. Most mutations are so tiny in their effect that they can be compared to a single pea under the many mattresses. The mattresses are the levels of complexity which separate the whole organism from any single nucleotide change in the genome. The more complex an organism is, the more pronounced is this phenomenon. Sanford writes, the primary axiom does not just ask the princess to sense if there is a single pea beneath her stack of mattresses. Through these mattresses, she must feel encyclopedias written in molecule-sized braille bumps and decide which volumes have the fewest mistakes. If we go back to our red wagon analogy, remember that the judge does not look at the manual, only the finished product. That is what is selected. If we're going to imagine this, imagine one single point mutation as the handle coming off of the door of one locker in the men's locker room of one of many fitness centers located in this large space station. Will the judge notice this as it looks at how the spaceship or space station functions overall in relation to other space stations? I mean, I think obviously not, right? There would be no way. Next topic here is noise. So this is something in Dr. Sanford's book. And when he talks about biological noise, he means other factors at play in survival and reproduction besides genetics. These non-genetic factors serve to confound the effects of genetic mutations. Environmental factors can give an unfair advantage to otherwise genetically degraded individuals, or they could wipe out individuals who possess beneficial mutations in spite of their benefits. These effects, both positive and negative, serve to drown out the signal of all but the most impactful of mutations. And since mutations happen individually and not according to any design or forethought, they can also act as noise to one another. A beneficial mutation could cause individuals to outcompete, even though they also contain many damaging mutations, and vice versa. And this means that there is ultimately no hope for natural selection to effectively weed out all of the bad and preserve all of the good mutations. And since these two types are vastly uneven, the majority are damaging, this also serves to ensure eventual decline. So if you look at these two red wagons, the one on the right side here has a lot of highly beneficial gain of function mutations with respect to the one on the left there. But nonetheless, the one on the right can still fall victim to being crushed by a giant boulder. And that would represent, that would be environmental noise, drowning out the signal of those beneficial gain of function mutations. All right. Now, we also need to cover an important topic, which is the word itself, fitness. We need to talk about the misleading way that evolutionists tend to use this term. And I've taken the following paragraph from an article I co-authored with Dr. Robert Carter some years back at creation.com/fitness. Fitness is defined in evolutionary biology as a relative measure of reproductive success of an organism in passing its genes to the next generation's gene pool. Evolution can be reduced to this one metric: has fitness increased or decreased? Now, a friend of this channel, Dr. Daniel Stern Cardinal, or as we frequently refer to him, just Dr. Dan, he's done us a favor recently in laying out very clearly the position he represents when it comes to this topic. He has written, deleterious means negatively impacts fitness, which means is selected against. And if you take the time to suss out what this quote actually means, he's essentially denying the existence of a class of mutations that are both deleterious and not selected against or unselectable. And that is manifestly not what the established experts in population genetics have to say on this topic, as we're about to see. But let's also talk about that word relative, because is real fitness truly only relative? Remember that life is like a very, very complex working machine. We've already heard from multiple genetics experts, both creationists and evolutionists, attesting to this obvious fact. So, with that in mind, imagine the gradual buildup of very small deleterious mutations, sort of like rust gradually forming on our red wagon over the years. Now, if we're only using this purely relative version of fitness, and we're going to look at this top left example here with respect to the other members of the population, we can say it has equal fitness. It's neither better nor worse than all the other members of the population, relatively speaking. But as that rust is gradually building at about the same average rate across all members of this population, look what we've got going on here. Even though all members of the population are clearly degrading, we actually still have the same fitness as before, relatively. And still, as this process progresses, our selected example has the same fitness as before because it hasn't changed relative to the other members of the population. But clearly, we need some way to describe what's happening here. Clearly, it's staying the same relative to the other members of the population, but it's not staying the same. It's degrading. In the following expert citations, it will become clear that this relative-only concept of fitness simply will not do. Now, in my previous debate on this topic, I used the following terminology. I'm not claiming that this terminology comes from any expert witness or source. This is just the terminology I used because I found it helpful to elucidate what we're talking about. I say, rather than just saying fitness, we need to clarify. Are we talking about relative fitness, which is the example that we already just discussed, or are we talking about absolute fitness? And that would refer to the functionality of the organism, irrespective of its reproductive ability relative to other individuals in the population. And I'd like to suggest what I think should be evolution's most important key performance indicator. For those of you in the corporate world, a KPI means key performance indicator. This is the most important thing we're looking at. And I think it should be for evolution, average absolute fitness over time, or AFOT. And after everything we've just been learning, what do you think that an AFOT curve might look like with biologically realistic parameters? I think, based on Dr. Sanford's research in his book, his AFOT curve would probably look a little bit like this. Gradual decline, and then you start to reach a tipping point where lots and lots of negative mutations are collectively building up, and you have something called synergistic epistasis, where they start to have a greater and greater collective impact, more so than just being additive. So you're going to start to get a really steep fall off, and what geneticists refer to that steep fall off, it's actually called mutational meltdown or error catastrophe. And when you reach that stage of degradation in genetics, you get extinction. So what we're looking at here is an extinction event. Now, going back to our expert witnesses from the secular academic world, we have Dr. Adam Iyer Walker, PhD molecular evolution, and Dr. Peter Kitely, PhD genetics. And they write, it seems unlikely that any mutation is truly neutral in the sense that it has no effect on fitness. All mutations must have some effect, even if that effect is vanishingly small. However, there is a class of mutations that we can term effectively neutral, the fate of which is largely determined by random genetic drift. And they continue, as such, the definition of neutrality is operational rather than functional. It depends on whether natural selection is effective on the mutation in the population, not solely on the effect of the mutation on fitness. Now, I've added that underlining because if what Dr. Dan said were the case, or if that is what these experts would agree with, then there's no way they could have written this, because they are clearly divorcing the concept of fitness effect from natural selection. Clearly, they are teaching that a mutation can be both deleterious and outside the realm of natural selection at the same time. And that is the definition of effectively neutral. So I'm throwing down a red flag here on this particular part. They are definitely being clear that you do have this class of mutation that we're discussing. They write, particularly for multicellular organisms, most mutations, even if they are deleterious, have such small effects that one cannot measure their fitness consequences. Now we're going back to one of the founding fathers, if you will, of neutral theory in population genetics, Dr. Moto Kimura, PhD, population genetics, from his paper in 1979 where he was laying out his model. Note that even if the frequency of strictly neutral mutations is 0 in the present model, that would be mutations that have absolutely 0 effect, a large fraction of mutations can be effectively neutral. Under the present model, effectively neutral, but in fact very slightly deleterious mutants accumulate continuously in every species. So effectively neutral because why? They are invisible to natural selection. But he contends they are still slightly deleterious. Now here's where it really starts to get good, because he says, whether such a small rate of deterioration in fitness constitutes a threat to the survival and welfare of the species, not to the individual, is a moot point. I'm going to stop right there, because let's think about this for a second. How can that be a moot point? The only way that this is a moot point, if you've got gradual genetic degeneration happening, the only way that could be moot is if you, like Dr. Kimura, are taking evolution for granted. You're not willing to question it. Because if we're willing to think critically about evolution, then this is the opposite of a moot point. This is a big deal. But he seems to intuitively understand that some type of rescuing device is needed here. It's actually not a moot point. He needs to explain how this degradation is going to be dealt with. And this is how he does it. He continues, but this will easily be taken care of by adaptive gene substitutions that must occur from time to time, say once every few hundred generations. I'm throwing a red flag down here on this underlined section, because clearly this type of language is the language of someone who is speculating. This is not something that Dr. Kimura witnessed or included at all in his model. If you look at his paper where he's showing his model, beneficial mutations aren't even included in the model. So this is not something that he's witnessed or modeled. This is a speculation that he's throwing out as a rescuing device to save the theory of evolution from what would otherwise be a very serious problem. And while we're on this quote, Kimura must have been referring to a decline, if you think about this, in absolute fitness, not relative fitness, since relative fitness by definition cannot decline across an entire population simultaneously. Relative fitness, or relative anything for that matter, is always a zero-sum game. So relatively speaking, just as we saw previously, all members are degrading at the same rate, well then relatively there's no difference happening between them. So if you have a degradation that is happening in all individuals of the population and it's a threat to the species, not to the individual, then you must be talking about absolute fitness. And here's my estimation of Kimura's AFOT curve based on his statements here. It seems like he's saying you've got this kind of jagged stair-step motion of absolute fitness over time, gradually going down due to the accumulation of near-neutral mutations, and then suddenly shooting up to a higher position than it was at originally through a mega beneficial mutation. And then the process just keeps going kind of like this. Now, Dr. Sanford has addressed the mega beneficial claim. He's written, the extreme asymmetry of mutational effects has to do with the fact that one is trying to climb Mount Improbable. Yes, it is conceivable that a mistake could cause you to stumble uphill, but only by a few feet. You will never stumble uphill by thousands of feet. However, a single error can easily cause you to fall downward very substantial distances. Indeed, while climbing Mount Improbable, you could easily plunge a very great distance to your instant death. Here's a little graphic I came up with to illustrate what we're talking about here. The mega beneficial mutations stumbling up Mount Improbable. Clearly, this is not how physics works, and it's not how probability works. He continues, in terms of a jet manual, a single misspelling might convert the command repeat loop 3 times to repeat loop 33 times. Or a misspelling might convert the command attach assembly 21 into body part A into attach assembly 21 into body part Z. These typographical errors could result in very profound changes in the shape of the airplane. But would they ever be beneficial? If they were beneficial, could they effectively offset the loss of information arising from millions of other misspellings, degrading all the other components of the plane? So here we have an example of a red wagon that is unfortunately very degraded. It has a high mutational load. It has rusted out and is clearly on its last legs. And then we get one of Kimura's mega beneficial mutations to the rescue. We just got a whole new wheel. Can you imagine that? Out of nowhere, a mega beneficial. We've got a whole new wheel. That's great, but what about the rest? Is getting a whole new wheel gonna do anything to help all of the other damage that has accumulated from very small, little deleterious mutations over a long period of time? Obviously not. Maybe we could even get a racing spoiler from another mega beneficial mutation later on. That would be wonderful. Highly unlikely, but great. But what about the rest of our red wagon? What are we going to do about that? And keep in mind, while we're waiting for these mega beneficial mutations to pop in, these overwhelmingly more numerous, very small deleterious mutations are continuing to accumulate. So this is just nonsense. It makes zero sense just on the surface of it. It's not even remotely plausible to think that mega beneficial mutations could outweigh the gradual accumulation of deleterious near neutrals. That's just not that. The only way you could think that way is if you're vastly oversimplifying the genome and life in general. But wait, because here's another issue. Kimura, later in life, became something of an apostate with respect to his own model that he promulgated previously. Evolutionary apologists will be quick to point this out, that Dr. Kimura made statements later in life, say like in the early 1990s, for example, that contradicted his earlier model. Now, modern-day population geneticists like Dr. Ira Walker have remained with Kimura's earlier published work, as that is what has proven consistent with the observable facts of genetics. And just to make sure I wasn't crazy about this, I personally contacted Dr. Ira Walker by email back in 2020 to ask him why he thought Kimura would have abandoned his own successful model. And here is the response that I got back from Dr. Ira Walker. He did give permission for me to share this. Despite being a co-author on the first paper with Tomoko Ota that proposed the theory of nearly neutral mutations, and written his 1979 paper in which he analyzes the model in some depth, he seems to have largely shied away from the model in his later work. I'm not sure why, but this might have been due to the fact that Tomoko Ota was the originator of this idea, and there might have been some professional jealousy involved. He goes on, I think it's been generally accepted that drift will tend to fix slightly deleterious mutations and as a consequence, fitness will tend to decline. It is also generally accepted that this can be compensated for by the fixation of advantageous mutations. I have attached one paper which has considered this decline, but this is by no means the only one. So what you see here is that Dr. Ira Walker is essentially saying the same thing that Kimura himself said back in 1979. He's saying, yes, overall fitness is on the decline because of slightly deleterious mutations, but then we're going to appeal to this rescuing device of mega beneficials to overcome the issue. And I did take the time to look at the paper that he attached, and it's a paper that he co-authored with Dr. Kitely as well as Dr. Martin J. Lurcher, a PhD in computational cell biology. And the paper had nothing to do with the actual mega beneficials. What their paper was about was showing the decline, or an example of this decline. We estimate that humans and chimpanzees have accumulated approximately 140,000 slightly deleterious mutations each, mutations that would have been eliminated by selection in murids. Murids are, I believe, like rodents. These mutations have small effects. And it should be noted that it is unlikely that the mutation accumulation is due to a recent relaxation of natural selection in humans due to an improvement in our living conditions, since the time of this improvement is short relative to the overall length of human evolution. So they're saying, no, this decline isn't because of modern medicine or anything like that. However, just like Kimura did, they see this as an issue, this is a challenge, so they're going to bring in a rescuing device. And here they say, we would not expect the decline in fitness to continue indefinitely, since the absolute strength of selection on new mutations, both advantageous and deleterious, may increase as fitness declines. So here comes our red flag out again, because again, we've got speculation happening here. Every time you try to look and figure out, okay, this paper isn't about this, the paper is only showing the decline. But then, they have to throw out this speculation. Well, maybe somehow an equilibrium is going to happen. Somehow selection is getting stronger as the degradation gets to a more critical point somehow. So if I'm trying to figure out what type of an AFOT curve Dr. Ira Walker et al. Would be showing, it seems like and I could be wrong here. I'm just trying to put the pieces together in what they're saying. They're saying that somehow, you've got the decline, but it reaches an equilibrium point, and then you get a mega beneficial to shoot it up past the starting point, and then it just keeps going. Although what I don't understand is how are you going to have different equilibrium points, on this chart? Like, so, this may not represent conceptually what they're trying to get at, but it's the best I could figure what they were trying to say. But it still doesn't make any sense, because not only, it just doesn't make sense. So furthermore, this accumulation of deleterious mutations may have been compensated in part by adaptive substitutions in gene expression control regions and elsewhere in the genome. So red flag out, again, may. They're using the speculative language, but they're kind of throwing both of these rescuing devices at us here. We've got the number one, mega beneficial mutations stumbling up Mount Improbable. And then number two, it's called sometimes the mutation count hypothesis or equilibrium. Mutations get more selectable as more and more mutations accumulate. So Dr. Sanford has responded. This is actually taken from the joint article that I co-authored with Dr. Sanford and Dr. Carter. He wrote, they have not carefully thought through what they are saying. The nature of near-neutral mutations is such that they are not only unselectable due to environmental noise, but they are also unselectable because they are noise to each other. Thus, as the number of neutral mutations accumulate, selection gets worse, not better. If each individual has 10,000 near neutrals, selection has to try and select for or against all 10,000 conflicting mutational fitness effects simultaneously. 10,000 independent mutational fitness effects usually bad ones, vanishingly few good, will not just be pulling in different directions with each other, but they will all act as noise, blotting out the fitness effects of each other. The claim of equilibrium has no mechanism to support it. That's the bottom line. The mere fact that lots of mutations have accumulated in a population will not magically cause it to stop being true that most mutations, the vast majority, are deleterious. There's no way to put the brakes on this degradation. It's not going to stop being true that it's easier to break a machine than to improve upon it by making undesigned, random, haphazard changes to it. So natural selection can only pick from the living members of the population, all of which are going to have new mutations each generation. There are no perfect scribes out there, to go back to our assembly line. Humans get approximately 100 new mutations each generation. Again, remember, at least 99 of those 100, probably 100 of those 100 are deleterious. So if you imagine going back to our population of wagons, if these little X's here represent deleterious mutations, and if we're going to assume for the sake of argument that these mutations are not nearly neutral but are in fact selectable, then which one are we going to pick here? Obviously, we're going to pick the top right one because it has fewer deleterious mutations. So that's natural selection, right? Well, let's speed this forward. Now we've got that selection happening. Let's go forward a generation. Uh-oh, what just happened? What just happened? More mutations happened. Which one are we going to pick? Well, still this one. But guess what? That one is worse now than it was in the previous generation. Why? Because mutations happen every generation. So clearly, there just simply is no explanation for how you just stop this process from running into the ground, basically. So in the end, this all boils down to one thing. We are doomed. Just as God said in Genesis, God cursed not only mankind, but all of creation as a result of man's sin. Our only hope for the future is not natural, it is supernatural. It is Jesus Christ. So with that, I want to direct you to these places where you can go for further reading. Obviously, I suggest you read Dr. Sanford's book, Genetic Entropy. Even many of his critics have not done that. But I also recommend you go to geneticentropy.org, which is Dr. Sanford's website, as well as creation.com/genetic entropy versus evolution, and creation.com/genetic entropy defense. And with that, I've reached the end of my presentation, but I do want to, before we move this now to the open mic section, I have, it's been laid on my heart all this week. For whatever reason, I feel that we, all of you who are watching this stream and are believers with me tonight, I really feel like we need to pray specifically for Dr. Daniel Stern Cardinal. And, he has been following this channel, and not even this channel, but just the general creation community for many years. And I've had many interactions with him. And I think he's a really smart guy. And for whatever reason, he's obviously very interested in the creation community. But I believe it's God's will that Dr. Stern Cardinal, or Dr. Dan, will abandon his skepticism and one day preach the faith that he has tried to destroy. I want to pray right now, Father, and I hope all of you in the chat and all of you who are believers will join me in this prayer that, Father, in Jesus' name, we pray blessings for Daniel. We pray that you will bless his life, that every good thing will come to him. But we also pray that you will reveal yourself to him in a way that only you can, that the scales will fall from his eyes, and that he will preach the faith that he once tried to destroy, just like another great Christian apologist from the first century by the name of Paul did. So Lord, in Jesus' name, we pray this. We pray blessings on Daniel tonight. And that was just something I felt that the Lord was leading me to do. So with that, I would like us to now move to the next section, Donnie.
Amen.
Paul, excellent presentation, very comprehensive. I love all of the citations, and which basically back the genetic entropy thesis. I mean, the founding fathers of population genetics and well-informed experts in the field of population genetics all agree that deleterious mutation accumulation is a real theoretical problem. Your correspondence with Kitely was revealing.
Dr. Ira Walker. Yeah.
Ira Walker.
Yeah.
That was revealing because, again, they are admitting to a real theoretical problem, but they are employing what I would say are rescue devices: adaptive substitution, synergistic epistasis, mutation count mechanism, junk DNA. But the point is, it is a real problem, or else they wouldn't have to invoke rescue mechanisms. If it didn't exist, then you don't have to invoke anything, right? It's just, well, that's not real, so let's not worry about it. But no, it is real. And the question is, are those rescue mechanisms plausible? So, Paul, excellent job, brother.
Thanks,
Sam,
guys.
any thoughts?
Yeah, no, that was fantastic. The slides and everything was very thorough.
I think it's one of the most
critics that join will have taken the time to listen to all of that.
And that's what we really want from the critics. So the join link is posted in the live chat. And so I've already got several backstage. Let's try and do our best to engage Paul's points in his presentation. But obviously the big question tonight is genetic entropy and the mystery of the genome. So we're looking forward.
And like I said at the beginning, I'm not an expert in genetics. So, there are people that are going to be able to say, well, what about this? And don't you know about this? And the answer is probably no, because this isn't about me. And all I'm trying to do is spread what Dr. Sanford has shared in his book and show the basis for what he's saying. So, and just by doing that, I think we can make a big difference.
Yeah, and it's more than sharing what Dr. Sanford has come to the conclusion of. It's population geneticists that you've cited, and you cited plenty of them. So it seems that it's pretty well established.
It is. Yeah, there's really not a big debate about it. Like Dr. Ira Walker said, there is the degradation happening, but then it's always, well, but here's the rescuing device. Here's why it's not a problem. But good luck trying to find the molecular basis for that. Good luck trying to find anybody actually mathematically modeling this rescuing device. It's just kind of thrown there, tacked on at the end, right?
Okay, well, let's have a great conversation as mods and hosts. Sam and I, we want to make sure that this is a conversation-based open mic. No monologues, no filibustering. If questions are asked on both sides, we want to make sure that they're being answered. And so Sam and I will make sure that it is more of a free-flowing and organic discussion. Of course, if somebody wants to present a slide or something, Jackson Rowe, I know, is asking if he could present some slides. Yes, but what we'll try and do is limit any kind of response on either side to 2 minutes max. But we do want to make our best.
Yeah, we don't have time tonight for everybody to come do their own presentation. So,
right, exactly.
But let's see what questions I can maybe help to answer, and let's see what we can do here.
Let's have some fun. So we're going to bring in the first two people, and we've got plenty to keep us busy all night. So what I'm going to do is we're going to have two at a time. The person to the right, Dr. Zach Hancock, great to see you. Great to have you on the channel. The person on the right's on deck. The person to the left will be the one personally engaging. Okay, we'll try and keep each interaction to roughly 15 minutes just to make sure that we've got more than enough time for everybody to engage Paul on the topic. Sam and I, although we are hosts and moderators, we may, from time to time, provide our own thoughts and input. Okay, so with that, what we'll do is we'll start with Jackson Rowe. I have a strong feeling once Zach is up. We're going to get real technical and deep into this topic, which is a good thing. So, Jackson, therefore, let's start with you and introduce yourself briefly. I don't think you really need a strong introduction. You've been here many, many times. And so, Jackson, quick intro. How you doing tonight, and which point would you like to engage first?
All right, I'm Jackson Rowe, and I don't believe in genetic entropy. But anyway, that's the introduction I'll use. But I think Zach will probably do a better job at this than me, but I'll just break the ice.
You will believe in it after tonight, Jackson.
We'll see. But we'll start with some stuff Kimura said. He didn't talk about beneficial mutations, mostly for the fact that he was talking about his neutral theory of evolution, not the neutral and beneficial theory of evolution. But also he talked about nearly neutral compensatory mutations, which were nearly neutral deleterious mutations would counteract each other. He spoke about that. I guess that's the first point.
Okay, so your first point is compensatory mutations. Basically, are you saying that they would counterbalance the damage due to the influx of nearly neutral mutations?
Well, I'm just saying that's what Kimura said. Yes.
Okay, that's what Kimura said. Okay,
I presented on that. I mean, I acknowledge that is what he said. I acknowledge that.
Okay.
But I also explained how he didn't provide any evidence for that, and even without going into the realm of evidence, what he said doesn't even make sense conceptually. It's not even plausible. You know, it's like if somebody said you can stumble up a mountain, it's not even all that necessary to ask for evidence, because just as a concept, it's incoherent. So the vast majority of mutations are deleterious. That is, again, extremely well established. So what Dr. Kimura was talking about was an extremely rare event. Adaptive gene substitutions, I believe, was the term he used. It's an extremely rare event that somehow comes in, and in one fell swoop, it's going to erase all of the damage done for generations previously by accumulating deleterious near neutrals. So that's what he's saying. I understand that, but I don't understand how that's even coherent, let alone how there could be any evidence for that. He didn't bother to give us any.
All right. I guess we're doing Zach goes next then.
Sure thing.
Yeah, that's
good.
That's fine.
Yeah, I didn't know if you wanted to like have a back and forth for a while, and then me and Paul can have a back and forth for a while, Jackson. Or if you just wanted to ask.
It doesn't have to be that way. If that's all he had to say, you know, that's fine. We can move on. And
well, I have another point if we want to do it that way. Or
yeah. Go ahead and have some time.
Yeah, you're on deck right now, Jackson.
Yeah.
So.
Okay. Well, you said Sanford never saw a beneficial mutation in plants. Just a quick look at Google Scholar. There's herbicide resistance in water hemp and multiple herbicide resistance.
That's actually not what Dr. Sanford said. He didn't say that there was never a benefit. He said it was almost a complete failure. He said the vast majority of the time it failed, and eventually it was almost completely abandoned. But he did not say that there was never any example of an improvement from mutagenesis. He did not say that. In fact, in his book, he even lists an example, and I can't remember. I know it had to do with corn, but I can't remember the exact mutation he mentioned for the corn that they did get that was helpful. But it was a reductive beneficial mutation. It wasn't, it wasn't a gain of function mutation, but it was beneficial for human purposes.
Here's the thing about these: they're not deletions or anything. They're duplication mutations. These two I mentioned.
Okay. What is the relevance of that point? Like, why does it matter?
Well, I mean, you guys frequently say things like it doesn't add information, and depending on what you mean by information, I mean that's kind of information.
Yeah. Right, but a duplication doesn't add information. Just like if I start stuttering right now, and if I say every word twice, you're not getting any more information from me.
If you enunciate the second word differently, maybe it'll have a different meaning.
If I could ask a question, aren't gene duplications duplicating the deleterious mutations with it? Wouldn't that be
most
speeding
genetic,
up the degeneration process?
A gene duplication is generally not going to be nearly neutral. It's going to be very impactful most of the time. And so it would be selected against. It would be deleterious most of the time.
It would fall in line with the high impact beneficials, which are rare. Selection may be able to amplify those. Selection may be able to see your worst, most detrimental mutations. But again, it's those nearly neutral, those slightly deleterious mutations that are accumulating, Paul, unchecked.
Exactly. Yeah. Those are the ones that are really killing us gradually over time, not the super negative, immediately, fatal mutations. Those, remember Dr. Sanford said, when you're climbing Mount Improbable, you can easily make a mistake that causes you to fall to your instant death. So that would be a super deleterious mutation that is selected against. But what you're not going to do is stumble and fall up the mountain all the way up. But that seems to be what Kimura is suggesting. So that's why I'm saying it's implausible. It's incoherent.
Jackson, any thoughts on that?
Well, it kind of goes to my next point a little bit, that his rust analogy for cars. I mean, that would kind of work if humans all consistently had the same mutations. They have the same mutation rate, but not the same mutations. And you're comparing it like cars all have the same amount of rust, while humans don't all have the same deleterious mutations. So this analogy doesn't really work. And a lot of the analogies were kind of clunky in my opinion, but that was the one that stuck out to me.
Now, I really like the analogies, but the reason you're getting tripped up here is that having different deleterious mutations, that's like having different rust spots. We're not talking about the exact same mutations. We're talking about the overall mutational load. So yeah, you look at two cars, they're not going to have the exact same molecules of rust in the exact same location, but they are, all other things being equal, they're going to accumulate an overall rust load, if you will, at about the same rate. So that's what we're talking about here. We're not talking about individual mutations. We're talking about overall genetic load.
All right. The other thing I want to talk about is kind of some stuff Sanford said in one of his lectures. I don't know if you guys want to talk about that or not, but I have some pictures.
It depends on if it's relevant to anything I just presented or not. If it's
well,
relevant,
it's
then
about
yes.
genetic entropy.
Well, if it's relevant to anything that I presented tonight, then yes. If not, then no.
Well, yes, I think it is.
Okay.
Okay, let me bring it up.
As you're sharing screen, Jackson, consider Kimura's rescue device with these adaptive substitutions. Well, there's no counterbalance because you have many more deleterious mutations reaching fixation. Your beneficial ones are very rare. There's a lot of noise, as Paul has talked about. So oftentimes the beneficial mutations are just lost. They're never fixed. They're never amplified. And so once you get a deleterious mutation, a slightly deleterious mutation fixed, that means it's stuck in place. That's irreversible or irreparable damage in the genome. I see it now, Jackson. Go ahead. Right. If you want to respond to any of that, feel free to do so as well, but go ahead.
All right, I'll just talk about the graph, because he talked about, there are so exceedingly few beneficial mutations. On this graph, he draws it out as 5,000 generations as basically zero beneficial mutations, and basically just exponential, almost upward gain of deleterious mutations with no selection taking anything out.
Okay.
I find that a stretch. But the next thing, his drawing of the slight line of beneficial mutations, and a few outside here, but the line of beneficial mutations in a no selection zone doesn't make any sense, because wouldn't beneficial mutations by definition be selectable?
No.
And one last thing from his presentation, Muller's ratchet. This only applies to asexual populations that have no recombination.
Okay.
So that's
I think
I drew a
okay. I think there's something called Haldane's
I
ratchet
drew a
as
thing
well.
here, but I won't get into that for now.
But some of that ratchet stuff, basically a ratchet, what it's talking about is that you get linkage blocks in your genetic code. So, you can't just take out any piece of the genetic code. Some parts are connected physically to other parts in a way that can't be separated. So what happens is that, even
Right.
if you get a beneficial in this linkage block, you're going to have potentially deleterious mutations that are also along for the ride on that linkage block. So it's just
Right, but
more
they
evidence that
counteract
natural
each other.
selection. So it's just more evidence that natural selection is not powerful enough to weed out all of the bad and preserve all of the good, like evolution essentially needs it to be able to do.
But when there's a beneficial mutation at that linked site, like you said, they counteract each other. That's the point.
Well, even if that were true, that's still not any forward progress. But I think when you say counteract, that's oversimplified, because we're not just talking about, I know we talk about mutational load and we talk about, fitness as this thing, this substance, fitness. But in reality, we are talking about genetic information. So, a beneficial mutation doesn't actually counteract deleterious mutations, because they're two different things. It's, just like I showed with the red wagon that grabs, it gets a spoiler, but it doesn't change the fact that the rest of the wagon is rested out. Because that's just oversimplified. We're talking about actual traits here. We're not just talking about a substance called fitness. Does that make sense?
Right, but there are compensatory mutations that even at linked sites that will counteract something. You're not just adding a spoiler to a car and calling it better.
Yeah, I mean, I'm not denying that can happen in principle, but you have to keep in mind the big picture here. The vast majority of mutations overwhelmingly are deleterious. So if you're going to overcome this problem, you're going to have to figure out a way to get rid of the vast majority of mutations and preserve this fleetingly rare event called the beneficial mutation. You're going to have to find some mechanism for that, and that's what the Darwinists simply haven't done at this point.
Well, we've been talking about this for several nights about purifying selection on regions that code for proteins, and how that kind of preserves portions of the DNA from so-called degradation.
Yeah.
So, purifying selection, that's one thing.
Well, purifying selection is the same thing as natural selection. It's not a different thing.
Right, I know that.
So if it's selectable, then it's outside of, you showed that graph of the no selection zone.
Right. Mm-hmm.
So if it's being purified, then it's out of that range.
It's high impact.
Yeah.
So it's the slightly deleterious mutations that are accumulating, Jackson Rowe, that are invisible to purifying selection. I think a good example of this argument from compensatory mutations or beneficial mutations has to do with Lenski's experiment. What results when beneficial mutations are amplified? Well, in almost every single case associated with Lenski's experiment, genes were deleted, broken, downregulated, or even eliminated. And what we see with Lenski's bacteria now is they have shrunk in functional genome size. And so that's the best you're going to get with beneficial mutation accumulation, is shrinking functional genome sizes, in the sense that there's no real trade-off. You amplify a single beneficial mutation, but the rest of the genome is still degenerating. Would you agree with that, Paul? Is that an accurate assessment?
Yeah, absolutely.
Okay, here's the last point I'll make, and here's one of the problems I have with this. The mutations kind of have to be neutral or nearly neutral to accumulate, but once they become harmful, the organism dies. And that's not going to happen all at the same time for all the organisms, right? There's no way it would happen all at once. So this is going to fluctuate and reach an equilibrium, would it not?
Well, remember I used the term average, so we're not talking about the individuals, we're talking about the population. The
Right.
decline we're talking about is a decline in average fitness over time. We're not just talking about individuals having lower fitness. We're saying the whole population is declining in fitness. And I just mentioned that nobody's actually shown any mechanism for this equilibrium. So yeah, you can just say the word equilibrium if you want, but that's not helpful, right? That's not evidence for anything.
Well, I didn't find the analogies very convincing, but I guess I'll hand it over to Zach also.
Sure.
Now.
Okay, Jackson Rowe, excellent back and forth discussion. I appreciate your points. And so, Jackson, what I'm going to do is put you backstage. Zach Hancock will be up, and Andrew Cumming will be on deck. And we've still got a packed house backstage, so we'll keep rocking the open mic this way. And so, okay, firstly, Zach, great to have you on. If I'm not mistaken, you were on several months ago for one of our open mics with Professor David McQueen, but this is our first face-to-face meeting, and so it is a privilege to have you here. Why don't you, for anybody unfamiliar with you in the audience, a little bit about yourself, how you doing tonight, and then pick a point for you and Paul to engage.
Yeah. Hello, everyone. I'm Zach Hancock. I'm an evolutionary biologist at the University of Michigan. I'm a postdoctoral researcher, and my work is in spatial population genetics. So this is my favorite topic in creationism because it's like the most relevant to like what I actually do. And I've had conversations with both Paul and Donny. So I've been like looking forward to like actually having a live discussion with you about this. So yeah, I'm excited to be here and have this conversation. So I kind of want to start off with just a couple of clarifying points from your presentation, Paul, and then maybe hop into a particular line of questioning. So one of the things that you talk about is absolute fitness. And I just want to clarify that absolute fitness is actually a population genetic term, and it refers to the non-relative version of fitness, right? So if you take the absolute or the highest fitness individual in the population, and then you divide that value by all of the offspring that other individuals have that are less than that value, then you're relativizing it to 1, right? But absolute fitness, you just don't relativize it. And that way, the total number of offspring that you have is your absolute fitness. And this is actually really, really useful because it allows us to connect genotypic fitness to population growth rates. And in fact, there's a longstanding ecological principle that approximately the natural log of mean genotypic fitness is equal to the population growth rate. So we can actually derive fitness values from populations straight from the rate at which populations grow if we don't relativize fitness. So I want to make that clear because it's an important component in evolutionary theory that is like relevant to this conversation.
Well, I mean, it sounds like you're agreeing with what I presented there. You're agreeing that fitness is not purely relative and that we can parameterize fitness with some actual objective measurable function. So the function that you're choosing to use here is, of course, reproduction. And that is, if you want to look at it that way, I would argue that is only one of many of life's functions, correct? There are many things that we do besides reproducing, like, for example, having debates. So, yeah, what you said is correct, but the only thing I will say in response to that is that when evolutionists try to act like the only relevant function in life is reproduction, I would say that is a massive oversimplification. And
do you
especially
so I'm sorry, but do you think that we actually think that way? That the only so because we're talking about me, right? So do you think that I go around thinking the only relevant function is life and is reproduction? Like, obviously, that's not true, right?
No,
like,
but is there any other function besides reproduction that you are aware of that has ever been tied to absolute fitness?
No, because again, absolute fitness is an evolutionary measure. We're interested in the spread of alleles through populations, and so we need a way to measure the rate at which we expect them to spread, and that's what fitness is.
Yeah, I understand, but I'm just suggesting, and I'm not the only one. Dr. Sanford has suggested that, we could parameterize absolute fitness with more than just simply reproduction.
You'd
We
fail
can
to,
add-
you'd fail to make any predictions if you did that. You would completely fail to predict allele frequency changes. You would fail to predict phenotypic changes through time because none of those other things contribute to that. So you'd just be muddying your model with no additional information.
I don't agree, based on the fact that if you just think about, let's think about, for example, breathing, right? So you could, and this is something that would be, I'm not suggesting this would be easy to do by any means. But I am suggesting that if you think about all of the many, many functions that are built into a complex life form like a human being, reproduction is, so for example, like I said, breathing, okay? When we breathe, we get a certain amount of usable oxygen out of the air. So imagine for a moment that deleterious mutations did something to slightly decrease the efficiency with which we could pull what we need out of the air. Just like when you go up really, really high and there's not enough oxygen in the atmosphere, that has an effect on you. Well, imagine a deleterious mutation that slightly decreased our ability to respirate, but not enough that it prevents us from actually living. I'm talking about very, very slight.
But that's still your absolute fitness. It's still affecting your reproductive fitness.
Well, no, because this
Yes.
this slight decrease in breathing is not going to affect your ability to reproduce in any way, shape, or form. Okay, because it's so slight.
Well,
That's
but
why
a very
that's why it's
slightly deleterious
unselectable.
mutation still affects your ability to reproduce. It's just the effect of it is very, very small. That's what we mean when we say an effectively neutral, though strictly speaking, deleterious allele, is there isn't a measurable effect on fitness. It's just so small that the rate of genetic drift overcomes it, right? Like the variance in allele frequencies overcomes the selective coefficient. That's what effective neutrality means.
I get that's the theory, but it seems intuitively obvious to me that if you tweaked my body's, like I have asthma, so I'm essentially one of these people that has a deleterious mutation that affects my breathing. I have asthma. So, but it's not affecting my ability to reproduce. And so what I'm trying to get across to you is that, yes, there are deleterious mutations that are degrading a function of life, but are not in any way impairing your reproductive ability. That's just intuitively obvious that that's the case.
But again, your argument is that that the accumulation of each of these effectively neutral, though strictly speaking, deleterious alleles will degrade. So that means that they must indeed, each individual one, have some measurable effect on fitness, even if very small.
No, it won't be measurable at all because, again, it's not affecting my ability to reproduce. So there's no measurable effect. It's not just that it's a very
Can you tell me what effective neutrality means? What is that? What you use that in your presentation. Can you tell me what that actually means?
So effective neutrality is it means that it does negatively impact. Again, it's going to have to be absolute fitness here. But
the way I defined that, like the actual way absolute fitness is defined.
Yeah, well, I think there is an inherent problem there that I'm trying to. That's what I'm trying to say. I think there's an inherent tension that I'm bringing out right now in these writings, because on the one hand, yes, they acknowledge there is a decline, and they acknowledge that all mutations must have some effect. But on the other hand, it's really obvious that not all mutations have any effect whatsoever on reproduction.
Well, if there's no effect whatsoever, then the distribution of fitness effects is zero, right? If the fitness effect has no effect whatsoever, that is a strictly neutral mutation.
With respect to reproduction, yeah. So I'm saying that is a tension that we're seeing here in the writings that I've chosen.
But none of this has to do with effective neutrality. That's my whole point. Is we're still not actually pinned down what that means.
Is absolute fitness, Paul, is absolute fitness equivalent to total functionality?
Yeah, total functionality.
And so total functionality is down. Although,
let's take a very simple example. Let's say you have an organism with 100 functions, and 99 of those functions represent your absolute fitness that don't contribute anything to the next generation. If only one of them does, then the next generation doesn't matter. 99% of those functions are completely lost. The only thing that matters is what is contributing to whether that organism is going to pass those traits on. If they don't pass those traits on, or they don't contribute to those traits being passed on, we don't care if they change over time.
Well,
right?
we do care.
It's only about whether or not, evolutionarily is what I'm talking about here. Evolutionarily,
Well,
and for like fitness in a genetic entropy sense, like, are we going to go extinct? That's about whether or not the population is going to collapse due to the accumulation of mutations, which means that it's going to decrease reproductive output. That is what that means.
Yeah, you're correct in the end. You're jumping to the end point here, but I'm talking about where we're like at this point. Like, I have a deleterious mutation, and my ability to breathe is just slightly less than optimal.
So
but it's not affecting.
Does it have an effect on your ability to reproduce?
Zero effect on my ability to
reproduce. It's strictly neutral mutation. So when you look at Kimura's curve, it sits at exactly strictly neutral, and it will not degrade population.
Kimura, in his own words, says that his model has 0 strictly neutral mutations.
I'm telling you, what you just defined is a strictly neutral mutation. It's one that has no fitness effect.
And I'm telling you, I agree, but at the same time, it's not. And there's a reason why everybody in this population genetics field is saying, look, there are no strictly neutral mutations, but they're missing one mark here. And the mark that they're missing is the life functions of an organism are all important to reproduction, but
then they're all contributing to fitness.
Well, I'm trying to explain this to you.
Okay.
I have a slightly decreased ability to respirate, but my reproduction is not affected, but that means that this damage that I possess in my genome is going to be passed along.
But if it doesn't damage you and it doesn't damage your offspring, then it's strictly neutral, and it will not damage the population.
You're saying that there's no difference between someone who has asthma and someone who has perfectly optimal breathing.
I'm saying what you said. That's what you just said. You said that your fitness is not impacted. You are still having offspring just as well as somebody with asthma.
But this is
but his offspring. I just want to point this out, Zach. His offspring, his progeny, are going to have more deleterious mutations than he did, and his grandparents did.
This is what I'm getting to.
I just want to say this. So the whole point. So the point of genetic entropy, as Paul is describing, is that every generation is more degenerate or more mutant than the previous generation.
But that means that it must actually be a deleterious allele. That's my whole point, is that if he's defining it as strictly neutral, then it doesn't matter how many of them they're not accumulating. Doesn't matter. The fitness effect is 0.
So if my offspring breathe slightly worse than me, is that good or bad? Is that a deleterious or strictly neutral mutation?
Well, we're bringing in some values in the good and bad. What we're interested in is, does it affect your ability to reproduce? Now, I'm fine with like saying that it does and considering it as like a effectively neutral mutation. That's fine with me. But let's nail down what that term actually means, because I feel like we're confused on what effective neutrality is.
Well,
because if it's strictly neutral in like the way you're defining fitness, it doesn't matter in an evolutionary sense. Populations will persist forever. It needs to be effectively neutral in the way I'm talking about fitness for it to actually impact population like function and growth rates.
Well, it's the aggregate that where it does impact. So yeah, in the short term, in this one generation, the damage to my respiration is strictly neutral with respect only to reproduction, but it doesn't change the fact that damage has occurred. And when you pass that damage on to the next generation, and then you add more damage onto it, eventually you will reach a point where it will affect reproduction. You know, if I had, instead of a 1%, reduction in breathing, if my breathing was reduced, if we just keep iterating, if my breathing got reduced by 75%, then yeah, you better believe that's going to affect reproduction. So you can't just say that in one generation, if something doesn't affect reproduction, then it doesn't matter. You can just throw that away.
I'm just repeating what you said. You said that it doesn't affect your fitness. So if you pass it on, it doesn't affect your offspring's fitness.
Zach, let me try to clear up, and maybe I'll try to introduce a different analogy, because it seems like you're kind of talking past each other. So imagine that each generation inherits dad's backpack full of rocks, but they're going to add one more rock. In the sense that you're talking about, in a reproductive sense, that backpack that's getting heavier each generation isn't going to prevent that organism or that individual from reproducing.
But this is good, Sam, because this is a measurable thing.
Hold on, Zach. Let me get my thought out. Eventually, there's going to be so many rocks in that backpack that it will affect reproduction. So what Paul is doing is drawing a distinction between what you're calling nearly neutral and something that's neutral in an absolute sense. Something can be neutral in a reproductive sense, but not necessarily neutral in an absolute sense, because in an absolute sense, these mutations are accumulating like rocks in a backpack.
But again, if there's rocks in a backpack, they're measurable. So they're having some effect, even if that effect is vanishingly small. That's an effectively neutral mutation. That's what we mean. But if we say that it has no effect, then it is strictly neutral and it doesn't matter, right? So we need to distinguish those two things. And again, we still haven't talked about what we mean by effectively neutral. I would still really like that defined.
Okay, so Zach, effectively neutral, it's defined in the literature. And I agree with the definition in the literature, only with this one caveat, that they are oversimplifying the concept of fitness by suggesting that we should only parameterize fitness by one metric only, which is reproduction. And the reason why that's misguided is that there are many body functions that we all possess which can be degraded in very tiny amounts without affecting reproduction. But we can't call that strictly neutral, because to say that it's strictly neutral would imply that it didn't actually damage anything, which would be incorrect. So
you still not told me what effectively neutral means.
It means that it is not affecting. It is not a selectable difference. That's what it means. It's not selectable. It's invisible to natural selection.
But it does still have a fitness effect. It does still have some effect on fitness, even if very, very small.
Well, I think that's what you're talking about there is the tension here. It's the tension where these experts want to be able to say that there are no strictly neutral, because it's intuitively obvious that any undesigned haphazard change to a highly functional machine, which they've confirmed that's what life is, a highly functional, highly complex machine. It's intuitively obvious that you're not going to be able to randomly and haphazardly just go in and make changes to the design or the code, if you will, of that machine, and have it just absolutely do nothing at all. So that's why they're saying, look, it's going to do something, even if it's vanishingly small. But the tension,
they're defining it based on reproductive fitness.
I know the tension is coming out because they're only defining it in terms of reproductive fitness. And what I've just shown you here is that it's very, very clear that there are ways that you can damage something without damaging in the short term its ability to survive and reproduce.
But what is this benefiting your argument? If the argument, like you cite all of these population geneticists that they agree that effectively neutral mutations exist, they're defining it under the distribution of fitness effects, which means that it's affecting some component of reproduction. Why are you adding in this additional thing? Why don't we just talk about what they actually mean? And so I feel like we're muddying the water here by changing definitions of things and suggesting that effectively neutral means something that's different than what effectively neutral means to all the people that wrote about it. But we're going to cite all the people that wrote about it and just like misconstrue what they mean. I'm very confused why we're adding in these extra definitions.
In Paul's example about a reduction in breathing every single generation, how can that be considered strictly neutral if there is a level of reduction at play?
We're losing function.
I'm fine with that. I'm fine with it being an effectively neutral, though strictly speaking, deleterious allele. He's the one that said it's strictly neutral.
Okay,
So
Zach, we're not going to get anywhere just continuing to argue about whether we're going to parameterize fitness by reproduction or
But your
something else.
entire argument rests on this point, and this is why I'm trying to make it, is if the entire argument of genetic entropy rests on effective neutrality, it rests on the accumulation of effectively neutral, though strictly speaking deleterious alleles, we need to know what that means. We need to know what is that box size that he shows. We need to be able to say how big is that box. Right? Those things really, really matter for this discussion. And if we can't even agree on what effectively neutral is, then we can't agree what that box size is. That's why this is important to nail down.
Well, Dr. Sanford has never attempted to define the exact size of that box, because to do that would require a level of genetic understanding that no expert possesses.
No,
It's far too complex.
No, it doesn't. That's
So-
not, again, this is because we don't understand what effective neutrality means. You don't need any other understanding to draw where the bounds of the box are. Now, you
Zach,
may struggle to
see if
measure
I
each
can
one of the fitness of, hold on, you may struggle to measure each one of the fitness effects, especially that are very small, but knowing where the bounds of that box is, that's quite simple. We do that all the time.
Okay, can I ask you to explain this for me? Like, maybe this can get us past this barrier to something more productive. Which one of the average absolute fitness over time conceptual diagrams that I put up on the screen, which one would you agree with? Or none of them? And why?
You're talking about the zigzaggy ones?
Yeah. Average AFOT curve. Which AFOT curve are you gonna
It totally depends. It depends on the effective size of the population. It depends on things like environmental stochasticity. Many things are going to shape population fitness through time, including things like the selective landscape. There are lots of things. So I wouldn't draw just one simple thing. But if we're tired of arguing about this, I do have some things that I would like to move on to, and I don't want to take up all the time.
Well, no, I want to make sure we talk about what's actually
move
on topic.
Okay.
So
Yeah.
this is the important issue. You're suggesting you believe in evolution, and so you are suggesting that average absolute fitness over time can go up and up and up,
No.
rather than what Dr. Sanford says,
That's
that
not,
it's
I've
going
not
down
said
and
that.
down and down.
I have not said that.
But here's the problem. This is why we also have an issue here, because if we're only going to parameterize fitness in terms of just sheer reproduction alone, if that's your parameter of fitness, then the problem is, well, guess what? That would mean that human beings are less fit than bacteria, because bacteria reproduce way more than we do. Okay? So that's not what we're trying to explain. We're not trying to explain how many kids can we have, okay? We're trying to explain how you get from a single-celled organism to a human being in genetic terms. How do you get that extreme increase of function or absolute fitness? But again, it cannot be parameterized only as reproduction, because then you're going to turn it on its head and you're going to say single-celled organisms are more fit than multicellular organisms because they reproduce more in an absolute sense. So how are you going to explain to me how you get from a single-celled organism to a multicellular complex organism genetically? That's what I'm asking.
Let's take a second. And again, we're so confused on what fitness is. If a population is stable in size, let's say you go out in nature and you measure a population and it's stable at around 1,500 individuals. What's the fitness of that population over time?
Well, can you describe whether you mean relative or absolute fitness?
Absolute fitness.
Okay.
And the way that it's actually understood in population genetics.
You just said that it's stable over time.
Mm-hmm.
So that would mean that
population size is stable over time.
So, okay.
Reproductively stable?
Reproductively stable. So that would mean that
that
would mean it would be flatline in terms of absolute fitness, right?
That means that the absolute fitness is 1, right? Because each individual is on average replacing themselves. And that's what we see very often in populations in nature, that on average, populations have a fitness of 1. They're just keeping the population size stable, right? And so very generally, what we see is that populations are sitting on these adaptive peaks, right? When you enter in a new environment, the fitness goes down for everyone, and then they explore a new adaptive peak, they reach the top of that peak, and then they stabilize there. And when you stabilize on the top of an adaptive peak, any additional mutations are dragging you off of the peak. So when you're sitting on a peak, most mutations are deleterious. To maintain like just population stability at an adaptive peak, most mutations must be deleterious. And so like that's the entire point I'm trying to get to, is that evolutionary theory absolutely predicts this deleterious mutation rate. It absolutely predicts that it should be this case. And not only does it predict that it should be this case, we can show, if we can agree on what effective neutrality is, that you can substitute every single base pair in the human genome to an effectively neutral, though slightly deleterious mutation and still persist. So if we can nail these things down, I can demonstrate both of these principles mathematically.
Do you agree with the statement that the vast majority of mutations are deleterious?
I just said that, and I just explained
Well,
why that
no,
is.
because you actually said that if you're at an adaptive peak, then they're mostly deleterious. So you added a condition there that the literature I quoted did not add. And actually, you gave a different reason for it than the literature gave as well. So
What did
that's
the
why
literature give?
the reason the literature gave was that it is much easier to break a complex functional machine than to improve upon it.
Well, if you're at an adaptive peak, any change breaks it.
It's not, it has nothing to do whether you're at an adaptive peak or not. It has to do with a fundamental design or engineering principle that any machine is more likely to be damaged by unplanned or undesigned changes to the machine than it is to be improved by those changes. That's the explanation, and that's the reason for it. So even if you're not at an adaptive peak, it is still the case that the vast majority of mutations are deleterious.
Again, they must be to maintain a stable population. If they weren't, if let's say there was an equal distribution of beneficial and deleterious alleles, populations would explode in size. We would be neck deep in elephants right now. They have to be this way, or the population would literally explode. Like, again, I can show this mathematically, or I can just try to explain it verbally.
Zach, you still haven't given even a single response to anything in the actual argument. All you've done is
you have yet to define effective neutrality. Don't come at me with this.
Yeah, well, okay, this is the problem. I mean, you want to argue about definition so that you can distract away from the fact that you actually have no credible answer to this problem. Kimura said there was a degradation over time, okay? Now, let's set aside the issue of how we parameterize absolute fitness. Let's take for granted for a moment that we're just talking about reproduction, okay? Even so, Kimura said there was a very gradual decline over time, right? We on the same page so far?
And why did he say that, Paul? Why did Kimura say that?
He said it because the vast majority of mutations are too small in their effects to be weeded out by natural selection.
Is that the reason, or is it because he only modeled a distribution of fitness effects that included negative?
Well, he only modeled negative because you essentially can't even measure. Beneficial mutations are so rare as to be essentially unmeasurable.
So we have no distribution of fitness effects that have positive values in it.
The ones I've looked at have never included anything on their graph for positive.
Hold on, Zach. So just to clear up the confusion,
Kimura certainly didn't.
We could grant, for the sake of this discussion, that there are positive mutations, but that would be a separate topic. We're talking about the accumulation of harmful mutations that cannot be weeded out by natural selection.
Well, I mean, again, as I said, I'm fine to do the math with that and show you why that's wrong. But we would need to nail down what effective neutrality means, and it doesn't seem.
Then you wouldn't be dealing with Paul. You wouldn't be dealing with the argument that Paul is presenting at that point. You would be.
Paul is inventing terms that don't exist in population genetics. So I can't address his argument if he's just making up terms.
Well, it's hard to argue one way or another with over talking. So, but what I'm saying is you would be deviating from the argument that Paul has presented tonight, and that there is an accumulation, a gradual accumulation of mutations that are not removed by natural selection. And it didn't seem like you disagreed with that. It seemed like that you agreed with the population geneticist that Paul has cited, but put a caveat on there about being at some sort of peak.
No, but Paul doesn't agree with them. That's my whole point. I can't make their argument because Paul doesn't agree with them because he's redefining
Zach,
fitness.
what is the explanation that you would like to give for how we overcome the decline that Kimura has identified?
But Kimura didn't identify a decline. That's my whole point, is that's not what that paper says. In that paper, he is modeling a very specific model that includes effectively neutral mutations under selective constraint. That's literally the title of the paper. It's a mathematical model. The whole point of it is he wanted to see a couple of things. He wanted to see, what does heterozygosity look like in effectively neutral situations, and what should the rate of molecular evolution be in effectively neutral situations. That's all that he did. Now, he needed a distribution of fitness effects to test his model against, so he created one out of his head. It's just a theoretical construct because it doesn't matter. He just needed a distribution. And in 79, we didn't have very many good distribution of fitness effects. So he tested his model against it. Now, at the very end of it, he includes beneficial mutations. There's a whole section on slightly advantageous beneficial mutations, not mega beneficial, slightly advantageous ones. And he showed that when you include slightly advantageous mutations in a constant environment, populations explode in size. They explode in size because if you're sitting on an adaptive peak, and you get a constant supply of beneficial mutations, your fitness is just going to increase forever and we'll be neck deep in elephants. That doesn't happen because, again, you only, like, beneficial mutations only matter for evolution when you're shifting peaks.
Okay. So to go back to what you said, I just have to clarify. You're essentially trying to rewrite Kimura's paper and have it say something different than what
No,
he actually
I'm saying
said.
exactly what Kimura wrote.
All right, I'm
Kimura's
speaking
paper.
now.
Hold on, Zach. Let's let
we need
Paul
to give
get
me a
his
chance
thought
to
out.
speak uninterrupted.
Yeah.
So I'll just have to go back and reread what Kimura wrote. Note that even if the frequency of strictly neutral mutations is 0 in the present model, that means every mutation has an effect, just like Dr. Walker said. A large fraction of mutations can be effectively neutral. And here's where this is what he says that refutes what you said. Whether such a small rate of deterioration in fitness constitutes a threat to the survival and welfare of the species, not the individual, is a moot point. Now, it was really, I think, asinine and ridiculous that he would call that a moot point because he just has agreed and he has just confirmed that according to his model, so it's dishonest when you claim that he was only modeling deleterious mutations. That's not the case.
It is the case.
Well, hold on. It is not the case because he just agreed that according to his model, there is a deterioration in effect. So you're
In his
denying
model under his
don't
distribution
break
fitness
in.
effects.
Okay. What we need to do is like mute the mic for a moment so I can speak uninterrupted.
You're rambling
Zach,
on.
let's do this. I'll jump in real quick. If there's points that you disagree with that Paul is saying in his uninterrupted response, write them down. Once we throw it back to you, we'll give you more than enough time to address them, and vice versa as well. Paul, go ahead.
Yeah. So Dr. Kimura is definitely telling us that according to his model, and actually Tomoko Ota's model, there is a deterioration over time in fitness. So what Zach is saying here tonight is that no, there is no deterioration. It's just a theoretical mind experiment that Kimura did. But what he didn't mean, he certainly didn't mean what he said when he said that his model shows a gradual deterioration of fitness. So I would say that's dishonest. Now, it's true that this paper is from 1979, but it's also true that modern day population geneticists like Dr. Walker fully support and endorse this model and still use it in their own research. It's not correct to say that Kimura was deliberately ignoring beneficial mutations in his model. What is correct is that he didn't bother to try to include them in his model, but then at the very end of his paper, he invokes them as a rescuing device despite the fact that he didn't model them. So, that's intellectually dishonest in itself, because you've got a model where you're willing to put a number on it for the deleterious, but then you're going to just leave the beneficials off, but then invoke them to overcome the problems that your model just demonstrated. And that's what he does at the end of the paper. He tries to dismiss the issue as a moot point, which it isn't. And then he says it will easily be taken care of by adaptive gene substitutions that must occur from time to time. So that's speculative, but here's where it gets to mega beneficial. Zach wants to say that he didn't mean mega beneficial. He only meant slightly beneficial. But that actually cannot work, because we have to have something that is going to effectively counterbalance the overwhelmingly asymmetrical nature of the distribution of fitness effects. So if the beneficial mutation is only very slightly beneficial, then guess what? We got a problem. We got a problem that the vast majority of mutations that are happening each and every generation are deleterious. So if a very rare, infrequent, beneficial mutation is going to somehow counterbalance the weight of deleterious accumulation, then yeah, it is going to have to be mega beneficial. So that's why we're calling it gene substitutions, instead of maybe a single point mutation or whatever. The point is, he's definitely having to invoke a mega beneficial here to overcome the weight of the genetic load. And that's just something that Zach is trying to obscure. And I would just have to say that everything he said he's spent more time trying to argue terminology and obscure words than he has to actually present any viable mechanism to overcome Kimura's decline.
Okay, thank you,
So
Paul.
I also got to say, we do have a lot of people here, so this isn't a Paul versus Zach debate night, but if we want to give Zach any final words, let's move on to the next one. But Zach, do you have anything else that actually addresses Kimura's decline?
Go ahead, Zach.
So at the end of the paper, just before the discussion, there's a section titled Slightly Advantageous Mutations, where he explicitly models advantageous mutations. It is literally in the paper. He doesn't just make up this last thing. You've read one line and then you quoted it, probably because you saw it in Sanford's book. But if you actually go to the paper, he models explicitly beneficial mutations. They are in there. The reason
they're not on his distribution.
I was quiet. I let you talk. It's now my turn to give my closing remarks. They are not in his distribution of fitness effects. Because, again, if you had read the paper, you would know why. He doesn't include them because if you have a constant supply of beneficial mutations, even infinitesimally slightly ones, they fix at a rate so much higher than beneficial ones or than deleterious ones, because deleterious mutations can only fix by genetic drift. Beneficial ones fix by selection. So even if they are very, very rare, they fix so quickly that it made his population explode in size, so he disregarded them. And because in real life, like, we don't live in a constant environment, the environment's constantly changing. This permits these beneficial mutations to not cause populations to explode in size. All of this is in the paper. It's in the section of the paper where he talks about this explicitly. Yeah, so, I mean, just to close out, because I have been taking up a lot of time and I want to let other people come in here and talk. Again, as I said before, the issue of having a lot of deleterious mutations relative to the number of beneficial ones is exactly what we expect in evolutionary theory. If you had an equal amount of beneficial and deleterious, given that beneficial fix so fast, the population would explode in size. You have to have more deleterious mutations and then compensatory beneficial ones very rarely, and you will have fitness equilibrium. I'm happy to send you papers on this, Paul and Donny, and I'm be happy to sit down and do the math with you on this. But I just want to say thank you so much for your time. I've really enjoyed it, and I will let Andrew and the next people have their say.
I want to ask Zach a question before he leaves. Does Kimura actually show the distribution of rare beneficial mutations to nearly neutral mutations accumulating, Zach? Does he
No, he
show
doesn't
the
show a distribution, but he gives it as gamma distributed, which is exactly how he does the deleterious.
Okay, but Dr. John Sanford does show the distribution of beneficial mutations and nearly neutral mutations.
Well,
and he demonstrates that the vast majority of beneficial mutations will be unselectable. He also points out that when you do get a rare beneficial mutation, today we understand that a lot of the times they're reductive. And so that's why I think the key issue does come down to what we were talking about earlier. What is the best definition of fitness? Because if it's reproductive fitness, I find that to be far too narrow of a definition of fitness. We need a more broad definition of fitness, which I think is total functionality. And that's more real world, where we have different environments that organisms are entering and dealing with. Go ahead, Paul. I heard you saying something.
I mean, it's just unfortunate that Zach has done nothing but obfuscate, because, again, most of the mutations that are deleterious in Kimura's model are unselectable according to Kimura. So we can't say, well, yeah, we have to have these deleterious mutations to keep the population in check. When Zach says that, he's suggesting that these are selectable mutations. And that's exactly the opposite of what Kimura is talking about. He is talking about unselectable, which means they don't thin out the population. They don't kill individuals in the population. So this red herring that Zach is bringing in about keeping populations in check is completely irrelevant to this entire issue that we're talking about. We are talking about deleterious mutations that are unselectable and do not, affect the death. They don't thin out the population. They are free.
We don't know
to
what they mean
accumulate.
because we couldn't define effective neutrality. We don't know what that box means. We don't know what mutations fall under there because we can't decide what effective neutrality means.
Effectively neutral is just unselectable.
Means we can't actually model
it's
it.
just unselectable, Zach.
This is just going to be.
It's more than that. That's why it's so important. It's more than just what's selectable. That's the whole point I've been trying to make, is that if we don't know what it means, we won't understand why this model is so incorrect. But
so
again,
Zach, before we leave, I just want to jump in. I want to be fair before we move on to the next person. Give me your brief 30-second response to this. I'll even give you the last word, and then we'll bring in the next person. So where Dr. John Sanford does respond to this in pretty good detail. He says, Kimura does not show the beneficial distribution, which is essential to the question of net gain versus net loss. This is what I just pointed out. When I, Dr. Sanford, show the beneficial distribution, while Kimura did not do this, I suspect he would have drawn it much as I did. Anyone can see the problem. The vast majority of beneficial mutations will be unselectable. And then even if one is selectable, again, most of them, as rare as they are, turn out to be reductive on a genotypic level, like we see in Lenski's experiment. But I don't want to make too many points. So I don't see how what you're saying here, Zach, and I do appreciate it, or the solution, adaptive mutations or beneficial mutations, is going to counterbalance any damage or answer the bigger picture question. So feel free, Zach, 30 seconds if you'd like to speak to that.
Yeah, I mean, again, Kimura did model them. He didn't draw a picture, so there's not like a figure one picture, but he models them as gamma distributed with a mean lambda. It's in his section. So you don't need to put numbers into this. He just does it algebraically. And so you can see what the effect would be. And it turns out that the rate of molecular evolution becomes proportional to the population size, which means that the populations explode in size. So that's why he was like, okay, this is clearly unrealistic.
It's clearly unrealistic. And gamma distributed just tells us the shape. It doesn't tell us the relative size on the graph. So that's kind of a little bit more.
That's what the lambda parameter does. That's what I'm saying, though.
So are you suggesting that Kimura was saying that the beneficial mutations were exactly equal in in impact to the deleterious? Are you saying he put the same?
You don't have to put numbers. It's an algebraic expression. You can put whatever numbers in it you like. The problem is that the rate of molecular evolution becomes equal to the population size. When that happens, it doesn't matter what numbers you put in for gamma and lambda. The population will explode because they're proportional. That's a problem. So you can model it. That's why he said slightly advantageous. Doesn't matter how small that distribution is. But I'm gonna go now. I'm gonna let Dr. Duff talk. Thank you so much for your time. Yes. I've really enjoyed it, and see everybody later.
Okay, Zach, thank you very
Nice
much
talk.
for joining. Dr. Hancock, we'll have to do this again at some point in the future. Always a fun topic to discuss. Okay, so we'll put Luca on deck. I'm saving Grayson for last because I think that's going to be a lively exchange. So let's get individuals who we could say is their
and we're
first
gonna
time.
have to. I think with Zach, we kind of we maybe spent more time than we should.
Yes, we
on
gave
one
Zach
person.
bonus minutes. So from
Yeah,
here
so
on
let's-
out, no one else gets bonus minutes. People have
And I'm
wanted to see
sorry.
Zach engage Paul and the rest of us. So we gave the audience what they want in that respects. But from now on, we'll stick back to the format. And okay, so we got Dr. Joel Duff.
Hold on,
Your first time.
before
Sure.
we turn it over to, I did have I think 2 questions for Paul, because when I was saying that we could grant beneficial mutations, the reason why I said that is it seems like Dr. Sanford in his mathematical model, he allowed for beneficial mutations in Mendel's accountant, and it didn't do anything. And then the second point that I'd like for you to address also is I believe that is Haldane's dilemma, shows that these beneficial mutations aren't as abundant and occurring as Zach would have us believe.
Well, yeah, no, I would agree. Zach was spending a lot of time kind of obfuscating without actually dealing with the issue at hand. But at the end of the day, you're going to have to figure out how you're going to counterbalance the collective mutational load of gradually accumulating deleterious mutations that are deleterious, yet at the same time unselectable. And it seemed like the only thing Zach had to offer on that was to try to just say, well, we don't know what that means. Well, according to the literature, we do know what it means. They are deleterious and unselectable, which means their cumulative impact is going to grow over time. They are subject to drift, not natural selection. There is an accumulation. So let's move on to Dr. Duff.
Okay, Sam, thank you for the questions. Paul, appreciate the answers. Dr. Joel Duff, good to have you here on the Standing for Truth debate platform. So, for anyone unfamiliar with who you are, brief introduction. How you doing tonight? It really is great to have you here, even though we may disagree on the origins issues. I do appreciate your contribution to these topics. So, Joel, a little bit about yourself, and then pick a point to engage.
Pick a point. Oh boy. I'm Joel Duff. Great to be here. I teach biology. In fact, I'm teaching genetics right now. I'm not a population genetics expert in the sense that I do all the modeling that Zach does, but I'm certainly involved in evolutionary biology and have many students who do those types of projects. So I see that. And I really don't see any sense in sort of digging into that pop gen, the nitty gritty of the genetics of that, given what we've just witnessed with Zach and Paul. But I did think maybe I would give one shot at maybe trying to get at this problem of understanding fitness and this absolute fitness, because it seems like just talking past each other here. And so I'm going to give the example I put in the chat. So Paul, you provided the example. I think you started. I'm just going to run with it, even though I don't think it's the best example. But you mentioned that you're asthmatic, and you said, like, as a character, that's not stopping you from having children. So, but you would consider that a deleterious trait, or something that is probably not something you would prefer to have, right? So but what makes something deleterious? I mean, is it just your perception, or is it how would you define that as being deleterious or a bad allele if it doesn't affect how many children you have? Let's just start there, and then I have another sort of a way of thinking about this that might help us.
Yeah, and this is the kind of this is the rabbit trail here, because it's very, very hard, and you would have to have a way to actually parameterize all the functions of life. It's easy to parameterize. It's easy to look at reproduction because you can count offspring, or like for a virus, you can count things like lysis time and burst size. These are things that you can easily, just count and measure. But when it comes to something like respiration, yeah, there are ways to measure that too, but it's very multifaceted. So it would require a huge amount of work to figure out all of the parameters that are involved in respiration and how to create maybe like some type of an aggregate index, like a respiratory health index or something, you know. But the point is, we can look at just reproduction if you want, and that's obviously what the evolutionary literature does. But if you choose to go that route, and if you choose to ignore all the other functions in biology besides reproduction, then what you're going to get is a drastically oversimplified look at mutational effects. So yeah, you asked, how can I know that having asthma is worse than not having asthma? Well, in a scientific sense, you would have to measure like the volume of air in my lungs and compare like the average respiratory intake and compare that to a healthy individual with no asthma, and you would see a differential there, and you could start to parameterize that. And you know, if you just continue to get worse and worse, you would see your respiratory ability going down, and eventually you would start to see a correlation between respiration and reproduction, right? There would be a correlation there, right? But at the very beginning stages of that degradation process, you would not see that. You wouldn't see the relationship there in the data. The data wouldn't show the relationship. The only time the data would show the relationship was once your asthma got so bad that you're as I mean, imagine how bad your asthma would have to be to cause you not to be able to reproduce, right? You'd either be uh like bedridden for your whole life or just dead, right? So this is the point I'm getting at. If you just reduce everything down to this one metric of reproduction, then you're throwing out a lot of highly relevant data to the overall picture of life, right?
Okay, that helps. That helps me see your frame of mind here. Ultimately, what you did though was you boiled it down to reproduction, because you said at some point when it gets to the point where it actually affects reproduction, then it really is a negative mutation, right? It actually is negative.
It's
And
negative
so
the whole
I would
time.
also say we already have a parameter that actually does generalize in a beneficial way the effects, and that is reproduction. And so my example here is, let me ask you this: if you had 1 million offspring that were born with, say, an allele, let's make it simple for asthma. Let's say there's a single allele that has a strong effect on whether a person develops asthma or not. And so 1 million children are born with asthma, or the allele for asthma, and there's another 1 million that are born without that particular allele. Would you predict that if you looked at those 1 million individuals, how many, let's say they had, well, let me just ask you, would you predict that those two groups of 1 million people, if you followed them and then saw how many offspring they have, would you predict they have the exact same number of offspring, or at least statistically the same number of offspring, or would you predict that one group might have more offspring than the other?
Oh, we're getting an echo effect here. Why are we getting an echo?
Let's
I don't hear
see-
anything.
Paul,
I
let me
just
see
started
if
getting a bad echo. Okay, I think it got better.
Yeah, you're good.
Yeah, I mean, I would predict if there's so many factors at play there. Are we talking about just having asthma at all, or severe asthma? Again, because if you're just looking at everybody with asthma,
I'm just
that's
talking
gonna
about that
include-
one allele, where we're just focusing on the one allele. This 1 million people have this allele. This other population has 1 million allele. This other 1 million people has this particular allele. So we're looking at the difference between those two alleles. So the question is, does that allele make any difference? Because that's what we're really talking about. We're talking about mutations. We're talking about individual locations, right? Individual sites, right?
Mhm.
Individual substitutions. And then the question is, do those individual substitutions actually make, are they relevant? Do they make any sense? Right? Do they have an effect? Are they neutral? Are they negative? Are they beneficial? So the question you could ask yourself is, for those million that have that allele versus the million that don't, would there be a difference in their reproduction?
I mean, I don't think so, but again, that's so
Then it's neutral.
oversimplified. Yeah, but that's the problem. That's the point I'm getting
It's
at.
a neutral allele.
But there are no strictly neutral alleles according to the population genetics experts. So this is what I'm getting at. That there is an inherent tension between the fact that, yes, things can fail to affect reproduction. And so that should, in theory, if all you're looking at is reproduction, then yeah, theoretically that should be strictly neutral because it didn't have an impact on reproduction. And obviously, there are mistakes that you can introduce into one particular individual at one point in time that will not affect their reproduction. So from that point of view, there should be strictly neutral mutations with respect to reproduction. But you
I
have
agree.
to ask yourself, why are the experts saying that that is not the case? Because that is what they're saying. They're saying there is no such thing as a strictly neutral mutation. And you have to ask yourself, why are they saying that? Dr. Duff, why do you think they're saying that?
Because it has so little effect on fitness that genetic drift will overcome that. So selection isn't going to have a great effect on those. If they're nearly neutral, they have some tiny effect. You really can't change anything without having some effect. So you can change any base, even a base that's between genes and has that, and it will have some difference, right? It changes the amount of energy that you need to copy an A versus a T. There could be a slight difference in energy there. And so one person's spending a tiny bit more energy than another person. So technically it's not completely neutral, right?
But now you're talking about
There is no site that is like
Yeah,
no difference you can have between two individuals that won't be neutral. However, the vast majority of them will act neutral with respect to how they're passed down over time, because
Yeah.
genetic drift is going to overwhelm the forces.
But Dr. Duff, you just started talking about something other than reproduction. You just started talking about energy use. That is a function other than reproduction. Energy use. That's not
Energy use will end up
so
making a difference in reproduction. If it doesn't, then it's effectively
exactly what I'm
neutral.
saying. Exactly what I'm saying. It will end up making a difference in the long run, population wide, when you accumulate these things long enough. But okay, let's take it back to the original premises of the argument. Premise one, mutations are the source of the raw material needed for evolution. Agree or disagree?
Sure.
Okay, so premise one, we're good on. Premise two,
We're good. We're golden. Let's move on.
the vast majority of mutations are deleterious and vanishingly few are beneficial. Do you agree with premise two?
Yes, I'm not sure the subjective words of vanishing and the vast majority of deleterious. We'd have to talk about the different ranges of deleterious, but Zach explained exactly that, and he explained exactly why most variations are going to be negative, and why having only a few of them is what's necessary. He explained
Okay.
that the whole adaptive
so
thing is
I'm
really
just trying to
critical
go-
for you to understand. If you don't understand that, you don't understand these ratios. And
Okay, I'm just
the
trying to go
relationships
point by
things.
point. So I'm getting your agreement or disagreement with premise two. The vast majority of mutations are deleterious, and
It sounds like
conversely,
you're the one asking questions now, and really this is the time for us to ask questions of you, isn't it?
so you don't want to? Do you not want to answer my question then?
Well,
you-
I'm
are you-
asking
are you
whether
agreeing
we're the
yes
ones
or no?
being quizzed or whether we get an opportunity to ask you questions. That's what I'm
Well,
asking.
it's an open mic
both.
debate. It's both. It's a debate, so it goes both ways. Joel,
Okay,
Yeah, it is
let's
gonna
take
go
one more
both
minute,
ways.
because otherwise he's taking all my time. I don't want to spend too much time when I have a couple other points to make.
Oh, okay. I think we can go ahead and cut it off here if Dr. Duff doesn't want to engage, because I'm specifically asking. This is the whole argument, and if you feel like what you have to say doesn't have anything to do with these three premises, then I'm sorry. What you came here to say is off topic. So I'm asking you, premise two, agree or disagree? Vast majority of mutations are damaging, and conversely, almost no mutations are beneficial. Agree or disagree?
Almost no mutations are beneficial? No, I can't say that.
Okay. Well, do you agree that the vast majority are damaging and that comparatively a tiny minority would be
The vast
beneficial?
majority are neutral or are near neutral or deleterious, yes.
Well, I didn't ask that. I didn't ask if the vast majority are near neutral. I asked, are the vast majority deleterious? Do you agree with that or disagree with it?
Disagree then.
You disagree that the vast majority of mutations are deleterious.
Based on your definition of deleterious, yes.
My definition.
Yeah, I think so.
Okay.
But let's go on.
Okay, so you disagree with. Okay, let's go back to the statements that were made by the experts in population genetics, and I just want you to hear what I'm saying.
We're not going to go to that. I'll just say that Zach has a presentation coming up that I think you'll find very helpful in terms of working out these issues with
Okay.
Sanford. And I don't think you and I are going to be able to work those things out. So I don't think that discussion is necessary.
I do think it is, and I think it's unfortunate that you're not willing to answer these very direct questions that I'm asking.
I want to make one point because Joel, you admitted earlier that, yeah, these slightly deleterious or these nearly neutral mutations, they are unselectable and therefore subject to genetic drift. But a
I
small
didn't say they're
fraction.
unselectable. Yeah.
Well, if they're subject to genetic drift because they're unselectable, correct?
Yeah, if they were selectable, then we wouldn't be talking about drift. We'd be talking about selection.
Right. And so
they can be subject to both. I mean, even if something near neutral could be subject to selection, but genetic drift could be a stronger force having more impact on the future of those particular alleles. But it doesn't mean natural selection doesn't have any effect.
But if the future of
it's not operating.
If the future of those deleterious alleles is fixation, once they're stuck in place where every single individual has them, that's irreparable damage to the population.
No, it's not irreparable.
Yeah. If it's a slightly deleterious mutation that's now stuck in place in the population, that is irreversible damage unless you're going to invoke back mutations, but that's very rare. So how do you deal with that issue, Joel?
Did you talk about selective sweeps?
So your answer would be selective sweeps of.
So is that selection then? Natural selection?
Well, selection on beneficial alleles, but that are linked to entire chromosomes or large portions of chromosomes that will then remove deleterious alleles. Yeah. I mean, no deleterious allele has to survive. And genetic buildup will cause individuals to become less fit, and they will be removed from the environment, removed from the population, and therefore their alleles won't be represented in future generations. I mean,
Joel, if an entire
but
if
at
an
the end
entire
of the day,
I want to address your point. If an entire population has a deleterious allele that's been fixed, and then you invoke a selective sweep of a certain part of the population.
Yes. Yeah, I got you.
Okay. So how do you? So a selective sweep's not going to address the fact that the deleterious allele is fixed in the entire population. Let's say you invoked a selective sweep. Yeah. Somewhere in Europe. Okay, so you remove those individuals from the
and every individual has it. We would have never gotten to that point of everyone having it. I mean, the reason everyone has it is because it either was a bottleneck population, and so now all individuals have it, but it's not so deleterious. Obviously, we're alive, right?
Yeah, but it is deleterious, though.
It's nearly neutral, Joel. That's the point.
It's deleterious. You can't just pretend that it's not deleterious. It is deleterious.
Right. It's a deleterious allele that's now
the population's growing and continues to expand, and there's more individuals, and the species continues. It's not
it doesn't matter because every generation.
But deleterious compared to what? Relative to what? Deleterious relative to what? A perfect allele?
But that's the point.
Dr. Duff, let me try to ask a quick question, and maybe that'll kind of help bring some clarity to what the prior doctor was saying. Let's say that you have two populations, and their reproductive fitness at this point is exactly the same. One of them is just reproducing, and they just spend all the time at their pool all day. The other population is accumulating genetically inheritable diseases. It's not affecting the reproduction. Now, would you classify them in terms of fitness the same way? You've got one population, they've got thousands of genetically inheritable diseases, but their reproductive fitness is not changed yet. And then you have another population, they have none of
They're
these
negative.
diseases.
The reproductive fitness is changed. You can't say it hasn't
Well,
changed.
they're continuing to reproduce the same amount for the sake of this analogy. Would you classify their
Well, then your
fitness?
analogy is incorrect. It's not possible. If they're negative, if you're defining them as negative alleles, then you've defined it by the fact that they do not reproduce as well.
Okay. Well, here's the problem. So for the reason why I drew that distinction is because it seems like that's where the confusion is. And when we're talking about humanity, we have the medical journals are full of thousands of genetically inheritable diseases. And these diseases come about by the same mechanism that you're invoking for forward evolution. So because that we are still producing and we are still accumulating genetically inheritable diseases, it seems to us that the way that you are defining fitness, being specifically regarded around reproduction, I know you're alluding to saying that, well, you know, it is affected a little bit, but we're trying to draw a distinction between these two things. Humans are still reproducing. Humans are still accumulating genetically inheritable diseases. And I think that's where the confusion is. Go ahead.
I don't think there's anything to say that. That's just
Okay.
There's not much of a point there. There's no real point there.
That the medical journals are full of thousands and thousands of genetically inheritable diseases. That's not relevant. That's the argument.
Do you not see why? I mean, it's like,
yeah, it's not relevant because
not relevant
because they're only want to talk about reproduction. That's the whole problem.
Yeah, right.
But guys,
if I'm born with a mutation, if I'm born with a
reproduction.
mutation that removes my hand, I have no hands. That's deleterious. That's harmful. That's hurtful. But it doesn't stop the fact that I can still have 3 kids. I can have 5 kids, 10 kids, and whatever deleterious mutations I inherited from my parents, those are going to be passed on to my children, plus some.
Donny, if you removed one of your hands, right, that wouldn't affect your genome. And so that's not going to be passed down anyway. It's not like your kids are going to be lacking hands. So that's not really going to work. But what if there was a mutation that did cause you, like individuals, to not have one arm? You might say, well, they could still reproduce. But if you had 100,000 people with one arm and 100,000 people with 2 arms, it's very likely, almost surely, that those 100,000 people with one arm wouldn't reproduce as many as those who had 2 arms. And therefore, the one-arm individuals will become fewer and less common, and that allele will become less common in the population, and the ones with 2-arm allele will go up in the population. Right.
Yeah,
so individually,
arm...
you're talking about individuals. Individuals. I have 5 kids. That doesn't mean I'm the most fit, except that it means I have high fitness in the sense of the population sense. But you know, I'm passing down negative
alleles. This is kind of a red herring here because we're not talking about
having any offspring.
Yeah, we're not talking about losing arms. This is kind of off topic because we're talking about changes in the genome that have no measurable fitness consequences. That's what we're talking about.
I guess where do I see the genetic entropy? Because when I look at a dragonfly, the fossil record, and I look at it today, I see they look pretty much the same. They have a large genome. They should be accumulating millions of mutations because they have large genome and they reproduce very, very quickly. Are rabbits like super less efficient today than they were in the past? I mean, did they give birth to hundreds of individuals instead of a dozen in the past? Were they better rabbits in the past? I mean, where is the evidence of genetic entropy that we can actually see? I mean, I know you love observational science.
The medical journals.
When I observe the world, forget humans. You're predicting this genetic entropy is happening to all organisms, right?
Okay,
the mitochondrial genome is accumulating mutations, right? But I'd say the mitochondria are really, really efficient. I can't imagine the mitochondria I have being a whole lot more efficient than it is today, even though it is the product of millions of divisions that have occurred to get to my point. So where's the genetic entropy there?
So genetic entropy it affects complex multicellular life much more than it affects simple forms of life. And the faster that you reproduce and the more selection is happening, and the lesser of a mutation rate that you have per generation, All of these factors come into play. And so when you want to bring up examples like dragonflies and mice or rabbits or whatever, you know, I'm not saying that those things are not affected by genetic entropy. I'm not saying they're not. But the types of organisms that are the most affected are the more complicated multicellular organisms, like the mammals, like humans. I know they're talking about now that
But you can
cheetahs
sequence a genome from
cheetahs are
5,000
very
years
close
ago
to
and compare it to
extinction.
today's.
What's that?
You can take a horse genome from before the Ice Age, so you believe that's close after the flood, and you can compare it to today's horse. And do you see a difference in heterozygosity? Do you see a greater buildup of negative deleterious alleles? You know,
Well,
do you actually witness it? You can do the
that's the
comparison.
problem.
You can do it. You can go out and compare organisms that are 4,000 years old with ones that are alive today. So from the flood to the present, and you could ask the question, can I see the genetic entropy?
Well, it's not that simple actually, because we don't have. If we had a
That's
copy
pretty
of the,
simple. You could line the sequences up and compare them.
yeah, but we don't understand genetics enough to know how those sequences exactly relate to the eventual function. This goes back to the princess and the nucleotide paradox. So you're talking about comparing the P or the nucleotide changes. I'm talking about through the matrices, what is the effect of that on the end resulting organism? So, you know, yeah, you can look at an ancient genome if we find one, but what you're doing there is historical science, not operational. And what you're doing, the problem is we don't
Comparing
have.
a genome past and present, that's
Yeah, you can
observational.
compare the two, and I'm sure you will see differences because mutations have accumulated. So both creationists and evolutionists would predict that there would be differences in those two genomes. So just
But your
finding
prediction is something
differences.
different than just differences. There are specific types of differences.
Yeah, but the only way we would be able to, you know, we would need to see a living example of this ancient horse and then actually measure the various life functions of that horse objectively compared to a modern horse if you want to start talking about that issue. We don't have a copy of the original created genome of anything. We don't have the starting copy. So, you know, it's an exercise in speculation to try to do what you're suggesting there.
Donny,
But
I don't want to. I
I
know
do
I'm
have to
running out
ask
of
that we
time.
move
I don't
to
want to.
the.
I don't want to go on with this, but I think I have something that would be much more interesting to your viewers. That's, I mean, it's on topic, but it's not on this specific thing. That I think will be more useful than spinning our wheels here. Sorry.
Sure.
Did
Yeah.
you
If
hear
you
me?
had one more point or question you wanted to.
I've only got a few
Yeah.
more minutes, guys, so we got to
Okay.
move through these callers
I
if
can
we're
make
going
it
to
relatively
get to
quick. There's two things. One is Nathaniel Jeanson, right? And Answers in Genesis and other young Earth creationist organizations talk about, starting with this perfect genome. And the way they think of it is having a massive amount of heterozygosity, right? Don, you've talked about this a lot, right? So you start with a huge amount of heterozygosity, and then somehow it's getting sorted out, and so organisms are becoming like more homozygous, presumably. Now, I would say you're going to have a hard time finding you can find some examples of that, but in terms of a general pattern, that's going to be hard to show. Nonetheless, genetic entropy suggests that you're constantly introducing all these mutations. And so organisms should be becoming more and more and more and more heterozygous. Right? That should be a prediction from genetic entropy, and it should actually be counter what Nathaniel Jeanson's predicting. Right? The two don't seem to be in sync with each other. The second thing, and you don't have to respond to that, but the second thing I wonder is, there's another young Earth creationist model that is called mediated design. Right? This idea that how did we get all the different characteristics we get today when in the perfect original Gardenic creation, organisms might not have had all the different features they have today that are adapted to today's world. So where did adaptations come from? Like, where did characteristics come from that they didn't need in the garden? They didn't need to be adapted to a desert. They don't need to be adapted to carnivores and so forth. So one of the ideas is that there's hidden genetic potential. And I see this all the time in literature. Georgia Perb and others talk about hidden genetic potential. Hidden genetic potential is this mysterious, or sometimes not so mysterious, genes or switches that are encoded by God knowing about the fall and knowing that after the flood, the environment will be different. And so organisms need different characteristics in the future than they have now. And so God, knowing that, placed those characteristics in those organisms, but they're not expressed, right? So you need to flip the switch at some point. And so like, I mean, like maybe ICR thinks of that as
Joel, if you want to sum it up quickly, just because I appreciate what you're saying, but just because Paul's got limited time. So I understand what you're saying. You want to try and wrap it up maybe in
30 seconds.
Sure.
All right. So all of that, here's my problem with genetic entropy with respect to what other creationists are saying, is that if these mutations are randomly accumulating, then they should be randomly accumulating in genes that we're not using today, or they weren't using all the way up till the flood, in which case they should be knocking out those switches. How does God prevent, right? What's God's action? How does God act in terms of preventing all these hidden genes and hidden switches from being mutated before they ever get a chance to be used? In other words, it seems like genetic entropy is thwarting God's plan for the future expression of genomes. You need to keep the perfect genome and the perfect genes in order to be ready for the future, for future environments. Genetic entropy basically wipes out mediated design as a creationist model.
Well,
would you agree or disagree?
You would. I think you would be assuming that God is an omnipotent and God is an omniscient. Because if it's
really?
Hold on, we're getting confused here because the time scale of genetic entropy is very long.
Right.
We're talking about a very short time scale here between creation, flood, and modern day.
Not for fastly, not for organisms that regenerate very quickly. In this case,
well, Joel, I just want to make sure.
Hold on, let me answer that. The organisms that regenerate very quickly also have greater amounts of selection happening, and they also tend to have a lower. A lot of these have a lower mutation rate per generation because of how quickly they reproduce. But again, I have to cry foul here because Dr. Duff is wanting to take us down all of these different rabbit trails, and he wants us to answer his questions, but he point blank didn't want to answer mine, and we don't have time for this. We got to move on to the next caller, please.
Okay, I'm going to jump in. Joel, thank you very much for joining us. I'd have my own way of responding in detail to your questions. I think they're good questions. It's something that we need like 2 hours to go back and forth on. I'm happy to do it. I'm happy to do it at some point. I want to get back to the bigger picture question now, the question of genetic entropy and slightly deleterious mutation accumulation. So Joel, thank you so much for joining. We got 2 people left.
Seconds.
What's that, Joel?
15 seconds.
No more seconds. Please move on to the next caller.
You were going to so miss it.
I want to respect Paul's time. Joel, this has been fun. I've got 2 people on deck.
It would be even more fun in 15 more seconds, but never mind. You don't deserve it.
We don't have time for this.
Nice to finally meet you.
Okay, Joel, great to have you on and interact face to face. We'll have to have you on again in the future. I've got 2 more people who will not like me anymore if I don't give them their chance to engage Paul. So, all right, we got Grayson on deck. We got Luca Medugno up. And so, all right, here's our final 2 contestants for tonight's show on genetic entropy. So, Luca, you've been listening for the last 2.5 hours. 2.5 hours. Go ahead.
It will be fast because I'm not a biologist. I'm a chemist. So maybe if we talk about entropy, I know a thing or 2, but with biology, not so much. But I'm very, very lucky tonight. So I have a very good example for you. You were asking for an example to demonstrate that theory, that hypothesis, about genetic entropy. And I have a very, very good for you. You are familiar with the Otzi or the Iceman?
Sorry, what is the thing you're talking about? Is it an ape man?
The Iceman, Otzi. It's a mummy, a natural mummy.
OTZI.
Yes. And it was found in Italy some years ago in the 90s, and it's a very old man. It was
Yeah.
from 5,000 years ago, and we do have his descendants here in Italy. We did found them. So with these corpse, these poor Otzi and those people,
Yeah.
we can demonstrate your hypothesis, and how.
Okay, well, this is the same thing that Dr. Duff was talking about, finding an ancient horse or genome or whatever. First of all, there's a lot of difficulty in actually getting a complete reconstruction of an ancient genome by trying to pull all of that stuff out. Obviously, it's been degrading for many years, so you're not going to get, an exactly perfect, undegraded look at what that genome was when the person or the animal was alive. So that's one issue there. But then the other issue is that, again, we're looking at so many layers of complexity, and there's so much about the genome, the genome's functionality, that we're only beginning to understand. So to suggest that we could somehow line up the nucleotide sequences between an ancient and a modern genome and try to figure out which changes are deleterious and which changes are beneficial, there's just no practical way to achieve anything like that. The only way we can know whether a change is deleterious in operational science is that you have to actually measure the you have to measure the consequences of the mutation, right? But you can't do that in this kind of historical science type of a situation that you're bringing.
Can I say something? So I gave you a very good example because, first of all, Otzi is basically intact. It was so much intact that when found, they believed that was a guy just lost to the mountain. It's that well preserved. We know basically everything about him. We know his DNA. We know we know that he was sick 3 times in the last 6 months before his death. We know that much of that man, and we have his descendants. So we know everything. It's the perfect example for you.
Okay,
So
that's fine.
I can bet that you will study the subject. Someone will.
Sure,
And
even if we grant that we would be able to perfectly sequence the genome from Utsi,
we can.
even if we grant that, and I'm not able to know one way or another, can we or can't we? I don't know. But if we can, the problem remains that we don't have enough knowledge of how the genome works and how changes to the genome affect the end organism. If you can't actually measure what happens operationally, then it's an exercise in futility to try to line up these nucleotide sequences and try to say, well, okay, that change is negative, that change is positive. There's no way to do that.
And we'd have to sort through, from the creationist position, we'd have to sort through created alleles and alleles that through mutation.
How are you retroactively? Exactly.
Right.
How are you going to retroactively figure out whether something was a created allele or a mutation event? How are you going to know that in retrospect, right?
As creationists, the majority of DNA differences that separate species and also intraspecies variation would be through created nuclear heterozygosity. Now, in the last 4,500 years, some mutations have accumulated, and we could look at the ratio of common alleles versus rare alleles. Dr. Rob Carter talks about this. But like Paul is saying, yeah, if you were to compare one genetic sequence to another genetic sequence, we're going to have to figure out a way to determine what's created, what's through deleterious mutation accumulation. Luca, go ahead.
Let me get this. So even bringing you a 5,000 years old corpse, basically perfect corpse for that age, you cannot show me any sign of genetic entropy. It's
okay.
What are you saying?
Well, we'd have to look at the genome. We might find deleterious mutations there.
We don't, guys, the way that we know about genetic entropy is not the method that Luca is talking about. So it's really off topic. But Luca, thank you for your time. Let's move on to Grayson.
I'd like to quickly give an answer to something that Joel was saying. And in a way, Luca, I think a good way to see an example of long-term genetic degeneration or entropy in like humans, for example, right? We exist in 7 to 8 billion people, relatively large population, obviously not compared to bacteria and mice and so on. But if we were to look to some of your extinct people groups, I think looking at these highly inbred people groups like Neanderthals, like Naledi, Hobbit, I think their extinction, the inbreeding and the manifestation of so many recessive deleterious mutations is a good sneak preview into where we are going genetically as a species. Again, today we exist in a larger population than Neanderthals did, but inbreeding reveals the hidden reservoir of genetic mistakes. And you can see the entropic effects in populations like Neanderthals. They were 40% less fit than modern humans. Okay. So that's an example of where we are going long term, because eventually so many mutations accumulate. You have all these recessive mutations, you have identical letters, you have just problem after problem. Okay. So Paul, I don't know if you think that's a good answer or not, but I think it's
it is. It's just unfortunate, it's tough to deal with this topic because there are so many different angles and rabbit trails, and that's why I'm not trying to sound mean or anything, but I really have to insist that we talk about what I presented about, guys, because that's what I'm here to talk about. That's what we got to stay on that topic. So, but thank you for Luca for
yes,
coming in
it
with
was a
that.
pleasure, and I want to add that it will be wonderful to study on that subject. No, if we can trace what the perfect genome was in your model, we will be able to live like 9 centuries or so, and that will be very, very good, I think. So
it would be nice, but we will never recreate a perfect genome. That is God's job, not ours, and we cannot reverse engineer. We cannot reverse engineer God's design.
Okay, John, we're going to move to the next person because I don't want to be rushing.
It was a pleasure.
Grayson, I want to give him. He's been sitting backstage
almost
for an hour and a half.
I am so very, very late up here.
Thank you very much. Okay,
good
you
night.
get yourself some well-deserved sleep. Okay, so real quickly before we allow Grayson to engage now. Lorraine Drosophila, thanks for the question. What's the evidence your children are more mutated? A question for me. Well, the fact that the mutation rate is roughly 100 new mutations per person per generation, and the fact that my children have more mutations, more slightly deleterious mutations than I did, and I have more mutations than my parents did, and their parents, and so on and so forth. And so every generation has more deleterious mutations. That's the evidence. Yeah. So pretty basic. Okay.
So real quick, I want to touch on that.
No, come on.
I don't want to be rude to anyone, but I really want to speed this along. Can we please get Grayson the opportunity to speak right now, because we just don't have time for any additional content at this point.
It was a question for you about how we.
I wanted to come in and debunk what Paul was saying, but it's so great because what Paul just said to Luca debunked what Paul was saying. Paul said that we can't possibly know the fitness effects of all of these changes. Even if we're comparing Otzi's DNA to his modern descendants, we can't possibly know their fitness effects. But earlier, Paul was saying that they're all deleterious. So which one is it? Is it
the vast majority,
or are they all deleterious?
Well, the answer is that the way that we know that most mutations are deleterious isn't through this process that you're suggesting here. The way that we know it has nothing to do with looking at an old genome and comparing it letter by letter to a new genome. That's not how we determine fitness effects. That's not how anybody determines fitness effects. So it's a red herring.
So how do you determine that every single mutation has to be deleterious? How do you?
No, I didn't ever say that. What I said is that the vast majority are deleterious, and I didn't even say that. I took that from the literature.
So I'm going to take you. How do you determine that? Because you were saying that you can't
Hold on,
You can't
Grayson.
determine
We're gonna cut back the over talking, and you'll get
Yeah.
plenty of time as you always do.
Oh, I thought Paul was done speaking. Sorry.
No, I just want to, Grayson, I've tried to do this with everybody with limited success. Let's go point by point. Point one, agree or disagree. Mutations are the source of the variation that mutation acts upon. True or
Sure,
false?
agree.
Agree. Okay, point number two. The vast majority of mutations are deleterious. Agree or disagree?
They're effectively neutral.
That's not the question. The vast majority of mutations are deleterious. Agree or disagree?
No, because they're effectively neutral.
Okay, so I got to explain to you that effectively neutral does not mean that it's not deleterious. And I can
Well,
take you to...
This is the whole point where Zach was trying to get you to define effectively neutral, because you're using your own definition of effectively neutral, not the way it's used in population genetics.
That is not correct. Dr. Kimura writes, under the present model, effectively neutral, but in fact, very slightly deleterious mutants accumulate continuously in every species.
That's
Do
not
you
his
agree or
definition.
disagree?
Like,
Agree or disagree with that
I
statement?
agree with what Kimura said, but I disagree that
Okay.
he is defining what effectively neutral means in that context.
What is the difference between effectively neutral and strictly neutral?
We've been over this. You went over it with Zach for a long time. You went over it with Joel Duff. I mean, we really aren't wanting to rehash.
What is just tell me the difference.
Or do you just want to get to the point where you ask me, what is it that compensates for this accumulation of slightly deleterious mutations that we both agree happens? And my answer is compensatory mutations. Drift and
that's
selection
not the question that I'm asking you at
drift
this
and
point.
selection entirely deal with every issue you've brought up.
That's an assertion and not true.
Ask it again. Ask it again,
Grayson. Grayson, the question I'm asking you right now is, what is the difference between a strictly neutral mutation and an effectively neutral mutation?
Why don't you tell me? Because everyone's been asking you
I'm
that all
asking
night.
you to answer my question according to your knowledge. And if you don't know, just say, I don't know.
I don't know what your definition is. Why don't you tell us?
He's asking you a question, Grayson, on how you define those.
I'm
things.
asking Paul to give his definition, because that's what everyone has been asking him all night long.
But he gave his definition.
I've
Now he's
answered it a
wondering what
million
yours
times.
is.
Yeah.
He's given it many times. He said effectively neutral.
Okay.
Is his definition is unselectable but slightly deleterious.
Okay.
So what's your definition?
Strictly neutral has no impact on reproductive fitness.
But they also said that
Effectively
each
neutral.
mutation. Hold on. They also both
Wait.
and
No overtalking,
the other guy. Hold on,
Effectively
Grayson. They both
neutral. I'm
stated
trying
that
to
there's no
respond to the
such
question,
Grayson,
Donny. Can I respond
Grayson.
without Sam?
Well, Grayson, you did kind of
I
sit
don't
in
know what your
silence for
problem
half a
is.
second. Sam jumped in. Sam, you finish your thoughts. We'll throw it right back to you, Grayson. Go ahead.
Am I talking with Paul or
they
Sam?
made it.
All of
Grayson,
us.
calm down. So they made it
just would
clear
rather
that there
speak
is
with
no
Paul than
such
Sam.
thing. Yeah, so for the fourth time, they said that there's no such thing as a strictly neutral mutation as you had just defined it. They said that everything has a slight effect on reproduction, even if it's not immediate.
Yeah.
So I'm wondering if your
If I
definition
could
even
actually
exists.
answer the question to Paul, please. I'm not speaking with Sam. If
All right, let's.
the
Go
S
ahead.
value is less than, I think it's 1/2 or 1/4 the effective population size, the S value here being the selection coefficient, then it is effectively neutral. There you go.
I know.
There's the mathematical definition.
Yeah, that's the mathematical answer. But what is that? What does that do with respect to natural selection? What's the difference?
Man,
you
I
can
want
give
your
a
definition
mathematical.
now. I just gave you mine. I want yours.
Okay, I'm going to give you Dr. Ira Walker and Dr. Kitely's definition. The definition of neutrality is operational rather than functional. It depends on whether natural selection is effective on the mutation in the population, not solely on the effect of the mutation on fitness. So if you want a clear definition of effectively neutral, that's where they give it.
Yes.
They clearly state that an effectively neutral mutation is not, natural selection is not effective on that mutation. And a strictly neutral mutation has no impact. An effectively neutral mutation can either be deleterious or beneficial, but what makes it effectively neutral is that natural selection does not act on it. Do
I
you
totally
understand
agree
that?
with you, Paul. And it's a function of the effective population size. Okay, like what is effectively neutral varies depending on the population
Yeah, we
size.
agree, but that's not the question I'm asking. The question I was asking is, what is the difference between the two? It sounds like we're on the same page.
Yes, we are.
You agree
Move on.
that most mutations are deleterious, and you also agree that there is a class of mutations, and in fact, it's most mutations, according to Dr. Ira Walker, that are not under the effect of natural selection.
Why don't we just move on? Because you complained earlier about getting bogged down in debating
Okay,
definitions.
so what's your answer? Is mega beneficial mutations? Is that your solution?
No, just any beneficial mutations. Even Kimura's own calculations show that slightly beneficial mutations will do the trick. And compensatory mutations, by the way, are empirically demonstrated and observed. It's not just a theoretical thing.
Okay, if you have 1,000 slightly deleterious, and then let's just overestimate the ratio, because it's more like 1,000,000 to 1. Okay, let's just say it's 1,000 to 1, though. You have 1,000 slightly deleterious mutations, and then you have 1 to every 1,000 that are beneficial. Now, explain to me where your math is coming from that 1 can be greater than 1,000.
Because we're talking about its impact on fitness and in the population. So if you have all of those slightly deleterious mutations, fine, they're not going to be drifting to fixation very fast. But those slightly beneficial mutations will go to fixation much faster and more than compensate for the fitness effects. And this can be mathematically demonstrated too. And we can demonstrate, as Zach was alluding to earlier, mathematically, that if you changed every single base pair in the entire human genome, every single base pair, to be slightly deleterious, you still do not have any kind of genetic entropy effects. It's like, because of the effective population size versus the size of the human genome versus the selection coefficients involved, you can mathematically demonstrate that this doesn't result in like any catastrophic outcome for the human population.
Okay, so the reason why a beneficial mutation would fix more quickly, that would be if natural selection were acting on it.
Sure.
But most mutations are not of that type. That's what neutral theory is all about. So
Beneficial
again,
mutations are.
you're sort of still implicitly applying to it, or you are appealing to natural selection here when you say that the beneficials will get fixed more quickly than the deleterious.
Yeah.
Well, no, they won't if they are effectively neutral. Now, if they're selectable,
they're not beneficial if they're effectively neutral.
But
that's
that's
like
the
a
whole
category
point.
error.
Effectively neutral is not a separate category from either beneficial or deleterious. That's what we've just established.
If
Effectively
it's
neutral,
beneficial, it will have a positive effect on fitness and be selected for.
incorrect. You're denying the existence of effectively neutral mutations again. And this is the problem with this entire debate, is that all it ultimately boils down to is that we're just going to keep like gaining ground and agreeing on a definition and agreeing on a concept, and then 2 or 3 sentences later, you're just going to go right back on it and contradict yourself. And
Wow.
that's all anybody's been doing all night.
That's
to be
all
honest.
you did with Zach. I mean, Zach is a population geneticist who knows these definitions. He's trying to tell you what the definitions are in the field, and you're doing exactly what you just claimed I was doing. You're backtracking on all of these definitions,
Okay.
and you're even contrasting with Sanford.
Okay. Grayson, thank you for your time. And I don't think there's anything more we can do here on that. I would like to get to Snake Was Right if we could, because I'm running out of time myself. But thank you, Grayson, for engaging.
All right.
Okay, thank you, Grayson. So we are just 10 minutes away. So it's up to you, Paul. We are just 10 minutes away from the 3-hour mark. And, we've given the critics and the skeptics 3 hours to basically join. Dr. Dan just joined backstage. I don't know if you want to put him on deck or, we are coming to the 3-hour mark. So I don't know how you want to utilize the last 10 minutes.
I did pray for him, so I guess we got to let him in at least for a short interaction. But and then after that, I'll have to cut it off.
That's
Okay.
cool.
So what we're going to do is
but I think Snake was right. Was right? So
let
I'll be
me
quick.
lay everything out. So we're going to stick to the format we've been doing. Dan is going on deck. So Dan, you just finished work. You probably didn't hear the introduction. So the person on the right's on deck. Person to the left, you being Snake, is the one engaging. And so we're going to make this the last two people, though, to everybody in the chat. We have appreciated everyone who has joined. It's been a great 3 hours. So, okay, Paul, and I appreciate your time, Paul, because you've been nonstop 3 hours. You also gave a nearly 1 hour presentation. So you got some great endurance, and I do appreciate it. Okay, Snake, what would you like to chat about in your time here? Good to see you, by the way.
Well, I would like to know what effect you think your prayer is going to have on Dan. What kind of brainwashing or taking of free will God is going to do on him. But I feel like that would be wasting my time here. But I really want to have that conversation with someone someday.
I'll tell you right now, I don't believe that my prayer will have any effect on Dan's free will whatsoever. Zero
I don't
effect.
think it will have any effect either. So I'm glad somebody finally brought up compensatory mutations, because I've been bringing them up for years and years, and creationists just kind of forget about them every time I come back to the conversation. And effectively, they're not the only thing that puts the brakes on slightly deleterious mutation accumulation. But they're a very demonstrable way of doing it. There are several experimental examples of scientists actually going in and damaging genes, and then these new mutations fix the genes and their fitness. And I can bring up examples.
That doesn't seem
we can
suspicious to
actually
you?
see it in action.
That doesn't seem suspicious at all to you?
No.
No? Do you think
What?
mutations are random?
Yeah.
And it doesn't seem
Actively?
suspicious to you that you're telling me that these scientists go in, they damage something, and then just by pure luck, it keeps getting repaired? Is that what you're telling me? That you don't find that at all strange?
So you think that they're manipulating it, or that by pure numbers? They've studied this. So there's about 9 different fixes of compensatory mutations that can fix a single deleterious mutation.
Yeah, but mutations aren't random. That's the problem here. They're not random. That is something we call
More or
them
less.
random because it's kind of a simplified way of speaking. But mutations don't happen. Mutations don't happen as a result of a die roll. They happen
Correct.
for actual
There's-
chemical reasons, right?
Right, there are hotspots.
And
I don't think
there's
there's any
hotspots
true
and there's
definition. I don't think there's any
Yeah.
such thing as true randomness.
exactly. So they're not
It's more of a
random.
mathematical randomness. So the point is, are you accusing these scientists of cooking the books?
No,
Essentially?
not at all.
Okay.
I'm just
So what's
suggesting
suspicious about it?
well, if you were playing cards with someone and you kept playing cards with them and they kept pulling out an ace every time, that might be suspicious, right? You might think that wasn't by chance. And all I'm saying here is, if you're speaking of a specific study, I don't know what study you're talking about, but if you're talking about a situation where mutations just keep getting fixed through a compensatory mutation, to me that sounds very suspicious, like something designed is happening there rather than an accidental mutation. And that's all I can really say, because we're
Well,
just
how
talking
about-
about you're talking about generic stuff. But the basic thing is here, you have to grapple with the fact, nobody has really grappled with it tonight, but you have to grapple with the fact that the vast majority of mutations are deleterious and they're also unselectable. That's what you have to grapple with. And you can just say compensatory all you want, but you've got to somehow find a way to get rid of most of the bad and amplify the very rare good in order to balance this equation. And I'm not hearing any mechanism for that here. Selection can't be the answer. So
well, it's a lot of, if there's like at least a dozen different selection methods. One of and on top of compensatory mutations. So Kimura did demonstrate that even with slightly deleterious mutations, the SDM accumulation is halted. How about we just grant that
No,
this
he didn't
compensatory
demonstrate that, though.
mechanism,
That's
well,
not correct.
mathematically in the algorithm, but let's grant that this compensatory mechanism is designed. It's irrelevant. If it exists, then it destroys the idea of genetic entropy.
Well, I don't believe that a compensatory mechanism exists that gets rid of all damaging mutations. That's not what I believe. That's obviously not true.
I don't think it gets rid of all damaging mutations either.
Well, okay, so what are I guess I don't understand.
There's not one mechanism that gets rid of all deleterious mutations. It's an accumulation of these several mechanisms.
Well, you listed selection, though, as one of your mechanisms, and that's off topic because we're talking about mutations that are unselectable. So that doesn't get to count as one of your mechanisms.
Well, because it's not genetic entropy unless it becomes selectable. Because
wrong. That's not what the literature says. You got to read the literature.
Well, the literature doesn't say anything about genetic entropy because it doesn't exist in the literature. It only exists.
Well, that's
in
like saying
circles.
that the Bible doesn't say anything about the Trinity.
Okay.
Just because the word genetic entropy isn't found in the literature doesn't mean that they don't say anything relevant to genetic entropy. Okay. Just doing a word search for genetic entropy isn't going to tell you if the literature is addressing genetic entropy or not.
So if it isn't impacting health enough to affect selection, then we don't have a scenario of genetic entropy because, okay, using your rust analogy, we're all slightly rusty, but unless it affects selection, which in the analogy would be parts falling off from rust, if we're only slightly rusty, doesn't matter. No organism, no biologist is going to say any organism ever was or is perfect. We all have imperfections, even well-adapted animals. It's not, there's no perfect fitness. So
it's the accumulation
of
it's the accumulation that counts. It's not the individual rust. I mean, I went over this and I don't know how I can explain it any better than I did, honestly, but yeah, it's the accumulation that's the problem. It's not the individual pieces of rust.
And if there's compensatory mutations in the analogy, we're having the rust constantly removed as more rust is constantly added.
But you would have
we have an
you would have to say
of just a tiny little bit of rust that's constantly adding, but constantly also being removed.
Okay, but for the same amount to be removed that was added, then you would have to be suggesting that the mutation rate is 50-50 balanced, and it's not.
No. Because you can have an overwhelming amount of deleterious or really slightly deleterious, and just a little bit of selection or a little bit of compensatory mutations is going to go a long way. That's where Kimura's model showed.
All right, you just appealed to selection again, and I've got to cut this off because we got to move on to Dr. Dan. So thank you though for your time on this.
All right, thanks, Snake. Maybe next time. Dan, you're muted. How are you? Welcome.
I'm great. How are all of you? It's been a long day. First week of the semester.
Yes, welcome.
But happy to be here talking about genetic entropy. I know Paul's been here forever, so I'm going to be really quick. I have one thing, and if we get to a second thing, then great, but we might not, and that's fine. But Paul, I'm going to be really quick. First thing is, it's not really a question. I just want to say thank you for refuting the Carter and Sanford 2012 H1N1 genetic entropy paper, a new look at an old virus. I think you're familiar with the paper. I think Donny and Sam know the paper I'm talking about. I forget the exact title. You know the paper I'm talking about, though?
Yeah, I know.
The 1918 H1N1 went extinct due to genetic entropy paper. And earlier you said that in order to characterize the fitness effects of mutations, you have to actually directly measure the fitness of them. You have to compare and see if they actually have a negative effect. And you specifically said that you can't just line up current sequences and ancient sequences and compare them and note the differences and make an evaluation, right? You said the exact phrase you used, I think, was exercise in futility, if that's your approach to determining whether deleterious mutations or beneficial mutations are occurring in those sequences. But that is exactly what Carter and Sanford did in that 2012 study, where they claimed they showed that H1N1 went extinct. They documented a bunch of differences in those viral sequences from 1918 to 2012. But one thing they pointedly did not do, and I emailed Rob Carter about this and directly asked him, and he confirmed that this is correct, they did not measure fitness in those viruses. The old ones, the new ones, none of them. At no point did they directly evaluate the fitness of those viruses. So according to the standard that you established just a little while ago, that study fails to demonstrate genetic entropy because they didn't directly measure fitness. Instead, they just compared the sequences, which, again, your words, exercise in futility. So my first thing is I just want to thank you for finally, after all these years, agreeing with me about that paper.
Yeah, and I'm not going to speak for Doctors Carter or Sanford, and I think you know, I know that you have written in on this, and they have actually given a response to you in the article, which is Genetic Entropy Defense at creation.com. And I'm not going to be able to do a better job of answering than they did. And at the end of the day, if I grant you, let's say for the argument that they made a mistake. I'm not saying they did make a mistake, but if they did make a mistake, that isn't really relevant to anything that I've presented tonight. And it's not relevant to anything Dr. Sanford wrote in his book either. So unfortunately, I'm not going to be able to stand here and defend their paper to you. Okay. I will say that I don't think, I'm not aware of anybody that has written in the published peer-reviewed literature to raise the objection that you're currently raising. So maybe that's something you can do, and then maybe they can respond in the literature. That would be appropriate, but it would be inappropriate for me to try to defend their paper because I'm not in that league to be able to do something like that.
That's fair. Second thing, and I promised I'd be really fast, so I'm gonna be very fast. Just a couple minutes ago, and I think earlier as I was listening on my drive home, I heard you say it as well, something to the effect of you're encouraging people to be familiar with the literature on this topic, right? Make sure you read the literature, know the background, so that when you talk about it, you're not, getting basic things wrong. I want to ask you if you're familiar with the Springman et al. 2010 paper. The title is Evolution at a High Imposed Mutation Rate. Adaptation Obscures the Load in Phage T7. Are you familiar with that paper? Because it is one of the most important publications on this topic.
Yeah, I'm really glad you brought that up. And I'm actually kind of surprised that you don't remember that I am familiar with it. Maybe you're just saying that for the sake of argument or whatever, but
I genuinely don't remember. Sorry.
Okay. It's actually something I wrote in response to you because you posted about this on Reddit. What was that now? 4 or 5 years ago?
Yeah, right?
I can't remember.
Yeah.
And I actually added a discussion of that article to my article I co-authored with Rob Carter at creation.com/fitness. I actually included a discussion of that article in that article that we wrote specifically because of you. So you have contributed.
Well, I'm flattered by it to hear that.
You have contributed to my work and more importantly to Dr. Rob Carter's work. So
can you refresh my memory on what the response was? Because that study is beautiful because of the fact that they weren't using a natural population. They mutagenized those phages, right? They hit them they were experiencing every mutation, right? So what was the response, if you could refresh my memory? Because I genuinely don't remember, and I apologize.
Well, okay. I'll quote a few relevant points here. The paper that you're referring to, it begins by saying, evolution at high mutation rates is expected to reduce population fitness deterministically by the accumulation of deleterious mutations. A high enough rate should even cause extinction, lethal mutagenesis, a principle motivating the clinical use of mutagenic drugs to treat viral infections.
Yeah, is that what happened in the study? Because that was the expectation going in. Is that what happened?
Yeah. So
no, it's not. The opposite.
I know. So I'm going to get to that because this paper, this is a weird paper, to be honest, and it's actually a confusing and self-contradictory paper. And I can tell you that behind the scenes, when I shared this paper with Rob Carter, that was also his reaction of this is just a flat out bad paper. And it's interesting. They claim that fitness increased.
It did.
And then they say a main plausible explanation for the fitness increase is adaptive evolution,
Mhm.
a process that lies outside the model. So right off the bat, they're admitting that adaptive evolution lies outside their model.
Their model
is-
of lethal mutagenesis. Yes, the
Exactly,
idea,
yeah.
exactly. So they were trying to model a process called lethal mutagenesis, where mutations accumulate and drive a population to extinction. And they go into some math in that paper, and adaptive evolution doesn't play a role in their model that they based their predictions on. But
Yeah.
when they did the experiment, which, I mean, I think when you do the experiment, you go by what the experiment shows rather than what your model shows, right? If the experiment shows your model's wrong, you change your model, right? So when they did the experiment, it contradicted their model, because what they found was the constant generation of extremely high fitness genotypes. The maximum fitness of those populations actually improved during that experiment, and at no point did those populations go extinct. And this refutes genetic entropy, because those populations, again, they were not natural populations, they were mutagenized. They experienced every possible mutation in those populations. And if Sanford is correct, if the vast majority of mutations are both harmful and non-selectable, let's put aside that that's a contradiction. I don't care. Let's give Sanford, let's let it go. If Sanford's model is correct, then a population experiencing every possible mutation will not be able to increase its fitness, right?
Well, this is the problem with the paper, is that they claimed that fitness increased.
They directly
But then
measured
when
it,
you
to be clear.
when you look at their actual measurements,
Mhm.
it contradicts their claim.
The maximum
So-
increased and the average decreased, which is consistent with the population genetics that we're talking about, that you find rare high fitness genotypes, but most genotypes are suboptimal compared to the ancestral
Well,
state.
there were two parameters for fitness in this paper: the lysis time and the burst size.
Mhm. Yep.
So when you read what they said, it says lysis time was difficult. So for those not familiar, lysis time would be the time it takes for the bacterium to burst and the viruses to come out, the new viruses.
Mhm.
So the lysis time went, it says lysis time was difficult to quantify precisely because phage release spanned several minutes. Lysis
Mhm.
time around 18 minutes and absorption rate were largely unchanged from initial values. Okay,
Right.
so lysis time was largely unchanged. So the amount of time it took the bacterium to burst didn't really change.
So the
But then
viruses
they
didn't
also-
get faster at killing the host necessarily, right?
So it didn't get faster at killing,
But
and they recorded an 80% drop in burst size, which means that
average.
that's fewer bacteriophages coming out on
Is
the
that
other end.
average burst size or maximum burst size? I'll tell you, it's average burst size. They recorded a drop in average, but they also recorded an increase in maximum. And
Okay,
that's
but
the key finding.
that means that the maximum was an outlier.
But that's okay, because we're talking about rare beneficial mutations, right? If we accept the premise that most mutations are harmful, then we would expect mutagenizing a population to, in general, decrease fitness. But if Sanford's model is correct, it should go into terminal fitness decline. And what actually happened is most of the population got worse. Some of that population got better, which means they found those rare beneficial mutations. If Sanford's model is correct, that finding is literally impossible. And that's why that paper, which is one of the few papers that actually shows a study of every mutation that's possible, right? We're talking mutagenizing these viruses. We're not looking at a natural population. We're artificially mutagenizing them to hit as much of the sequence space as possible, as quickly as possible. So according to Sanford, when you do that, because almost all mutations are harmful, and the vast majority of those are unselectable, so they're going to accumulate without any way to stop them, the population must go extinct in that scenario. But instead, what we saw,
and I can respond to that.
Maximum.
I can respond to that too. And so you're clinging to this like outlier that occurred and ignoring that the average fitness went way down. And it is bizarre. And I actually contacted the corresponding author for this, which is J.J. Bull.
Oh yeah, he's great.
And he said, having looked at the paper again, it is the case that we did not have an easy explanation for the increase in fitness. The burst size went down, so if lysis time stayed the same, there was a big variance complicating estimation of the mean, then fitness should have gone down.
But it went up.
What we suggest as a possibility is that the means of burst size and lysis time should not be used to parameterize the fitness model when there are large variances.
Yeah, a better.
But I'm sorry, that is the fitness model.
But no, that's wrong. The metric for viruses for how you measure fitness is called doubling time, which burst size and lysis time are parts of that, but you measure it empirically by doing a single generation and then diluting them to the point where you can actually count how many there are, and then doing the math based on the dilution. So like it's actually doubling time is the way you do the calculation. So the things that you asked Bull about are he's right in what he says, and it's consistent with what I'm saying. Yeah, that's that simple. Like, yes, that's what those words mean.
Yeah, I
my
understand.
point is, if I could just be clear though, my point is that Sanford's model predicts, and it must predict, that when you mutagenize a population to the point where you're sampling the totality of sequence space, right? You're inducing every possible mutation, right? That population is dead. It may be immediate, it may be in a few generations, but that population is doomed. That's what Sanford's model demands. That is, there's no way that can't be the case.
Well,
if Sanford is
not
correct.
every individual is going to possess every single possible mutation simultaneously, though.
But the population will decline, right?
Yeah, the population declines. Now, we did contact John Sanford for his take on this particular experiment. And, you know, what he stated, you said that, you know, why didn't we observe the mutational meltdown? Well, the simple answer is that they did not proceed long enough. Right. Because that wasn't the point of their experiment. They weren't trying to kill this population.
That's literally what they.
He was crystal clear.
Testing,
yeah, he was crystal clear that 200 generations is not long enough for the full effects of genetic entropy. Yep. It's not long enough to have actual mutational meltdown occurring.
Okay, so two things.
All they did was go 200 generations. That's not long enough.
Bull said that 200 generations is not long enough for genetic entropy. He said that?
No, Dr. Sanford said that.
Oh, okay, Sanford said that. Okay,
Dr.
the other
Sanford
thing is that
200 generations is not long enough for extinction to happen.
The other thing is that Sanford conflates two opposite things here when he talks about mutational meltdown. He does this in the book too, because mutational meltdown is the opposite of genetic entropy. It's when a population gets below the minimum viable population size and you have a loss of heterozygosity and an increase in recessive deleterious traits. So mutational, and he does this in the book as well. Mutational meltdown is the literal opposite process from genetic entropy, where genetic entropy is an increase in diversity, an increase in heterozygosity. Mutational meltdown is a loss of diversity, a loss of heterozygosity. So those are opposite things that I'm not sure that's clear, but yeah, Sanford makes that error in a couple places.
Yeah. Well, I am glad that you brought up this paper because, again, I know you were probably not able to watch the presentation because you were driving.
Yeah, I was at work. Yeah, sorry.
I talked about average absolute fitness over time. And that's what
and in that study it went up. The absolute went up. The max went up.
Not the average. So
no, but the
word average is important there. We can't just throw the word average out. Okay.
Population won't go extinct though if the maximum goes up.
Well, but it will when you can't say, oh well, we have one individual here that didn't have all those deleterious mutations, so it was doing good. Again, genetic entropy is about the population, and the actual evidence that we have from this paper shows an 80% reduction of burst size. That means essentially 80% fewer viruses being produced, and the lysis time didn't change. That's what happened here. That is genetic entropy.
But what
it's genetic entropy in action. You're losing.
Wait a minute. Hang on now, Paul. Hang on now. You got to be consistent. You got to be consistent, Paul. Did they measure the sequences? Did they actually do anything and show that these mutations cause this problem? Because the standard you established earlier, when I caught like just a snippet in the car, was looking at specific mutations and their fitness effects.
No, I was saying no. You can't. We don't determine genetic entropy by lining up specific nucleotides with each other
and testing their fitness effects.
That's not how we do it. But what we observe is that 80% drop of burst size over 200 generations with no change in lysis time.
But did the
fitness declined? It didn't increase.
No, they specifically say that maximum fitness increased.
If I'm a statistician, and I present my findings by ignoring the overall bulk of the evidence, and I only talk about the outlier that occurred, that is misleading and dishonest statistics.
So
and that's what they did here.
2 quick questions, because I feel like we're going in circles here, Paul. Just to be clear, the maximum fitness that they measured in that study went up, right? Just to be clear, the maximum fitness went up. If a population is undergoing its population genetics in such a way that there are some subset of individuals whose fitness goes up over time, is that population going to go extinct?
Yes.
Really?
Yes, because the average fitness for the population went down, and once you hit a certain point, you're dead.
What's
as a
the definition
population
for extinction?
Because that individual that you're talking about that had the higher fitness.
It was 1?
Yeah. Well, I don't know how many it was.
It was a lot. It was a lot.
Well, we're talking about viruses. We're talking
about bajillions, right? It's a lot.
Well, that's fine. But the average still declined by 80%. That's a huge drop.
But the population is still viable, and you can. And again, they did the math on it. You can do the math on it. Ask Sanford to do the population genetics math for you. Like that population
but you're
was
ignoring
not going
the fact
extinct. They went for
you're
200
ignoring the fact
viral
that the only thing they actually observed was decline in average fitness by 80% over 200 generations.
You keep saying the wrong words there, and I think you're doing it on purpose. They didn't observe the 80% was birth size. It wasn't fitness, right?
Well, I know, but there's
not
only
the same
two
thing.
components. There's only 2 components: lysis time, birth size. That's all they
one of them
used.
stayed the same, right?
One of them stayed the same. One of them dropped
by
So where's your
80%
increase?
on average. So if you put stay the same and drop on average by 80% together, do you get an 80% average drop?
Well,
No, it's the average
You're
drop in
equivocating between
fitness.
2 different parameters.
No,
You're equivocating
well-
between lysis time and birth size.
Yeah, because to get average fitness, you have to combine them, right? And you're taking the measure from one of them and saying that in its totality is fitness, right? You're saying
No.
the 80% drop in one means an 80% drop in fitness.
No, I
That's
don't
not
think that
true.
at all. The lysis
have to
time didn't
consider them.
change.
Exactly. But you just said an 80% drop in fitness, right? You
Well,
just said that.
All right, my mistake. An
Thank
80% drop in birth size. And let me tell you this, Dan. Out of everybody that's come on here tonight, and we had Zach Hancock and we had Joel Duff.
Oh, I missed Joel Duff, man. I missed so much fun while I was at work. This isn't fair.
But I have enjoyed this little brief a discussion that you're having with me right now infinitely more than I enjoyed anybody else that came
warms
tonight.
my heart
already.
so much.
Because I appreciate you being here, and I'm sure you're going to watch the presentation and make some kind of a slam video against me later, and I'm expecting that, and I won't be offended.
You know what? Just because you said that, I'm going to try. The new semester just started. I'm really busy, but just because you said that, I'm going to try. I'm going to make sure. I'm definitely going to watch it, but I'm going to try to do something with it.
Well,
so,
it's up to you, but I did pray for your I did pray for your salvation tonight, so I hope that comes through.
I appreciate that. Thank you, Paul.
Because if that happens, then you will become probably among the best creation scientists ever to live. That's my prediction.
I keep saying I should sell out. I could probably make more money if AIG hired me, because I could do better work than all they do.
Well, I don't know about the money part, but I know that if you applied your critical skills to tearing down evolution like you apply it to trying to tear down creation, I think we'd have something going on here. So
I appreciate that, but why would I do it for something that's wrong?
Okay, well.
Paul, this has been a lot of fun. I appreciate you staying late to talk to me. Donny and Sam, thanks for giving me the opportunity.
Our pleasure. Lots of fun.
Right,
that was a very good ending to tonight, and it ended on a high note.
I think Dan ended our last open mic with you too, Paul, if I'm not mistaken. Historical.
Did I always get here late?
No. The last time it was Mark.
Dan does it on purpose. He wants to be the main event. Actually,
closer.
behind the scenes, we all work it out that way. So. Yeah. This has been fun. Thanks for joining, Dr. Dan.
Thanks, everybody. Have a good night.
Okay, well, ladies and gentlemen, that brings us to about the 3.5 hour mark. Paul, excellent endurance. We had quite a few engaging back and forths. Dr. Zach Hancock joined, Dr. Joel Duff, Dr. Dan Stern Cardinal. So we can't say that the critics, including the PhDs, on the other side aren't engaging with genetic entropy. So that's good. This is what we want. We want engagement on both sides. We had Grayson join, Snake was right, Jackson Rowe, Luca Madugno. And so the whole crew was here tonight for what I believe was an excellent presentation, Paul, my brother, and also just an excellent open mic back and forth discussion. Hopefully this opens up more discussion and gets more people involved on both sides. So anyways, Sam, great job moderating, great job hosting. Paul, let's hand it to you. Do you have anything you want to add, anything you want to say? What were your overall thoughts in terms of the arguments being made tonight by the skeptics and critics? Do you feel like they've adequately addressed the bigger picture question that you presented today?
So yeah, I do want to thank Sam, Redefine Living, and Donny, Standing for Truth. Thank you both for being on with me tonight and kind of doing a little bit of tag teaming, but also, just keeping things in line to a certain extent. Anyway, we did kind of go off the rails at a couple points, but we recovered and we ended on a high note. And it's a lot of what happened is kind of what I expected, which is why this is a hard topic to debate, because it's very intricate and complex and so much nuance, and there are so many places where people can obfuscate and kind of equivocate in ways that are, it takes more time to clean up the mess than it does to make the mess, right? So that's the difficulty with this type of a debate. But I appreciate everybody who came on and participated. I think certain individuals were a little bit more on point than others. There are a lot of kind of obfuscation about definitions of words rather than actually addressing the meat of the argument.
Paul, if I could jump in for the audience, I want this to be as edifying as possible. And I felt edified. I restrained myself as much as I could. Like you said, Sam and I got involved here and there, but we mostly left it to the critics and you. So to make it as understandable for the audience, when the critics basically would ask you, what does population geneticists, leading population geneticists, what do they mean by effectively neutral? What were they trying to get out of that? What do the population geneticists mean by effectively neutral, Paul? And is it in agreement with what you and Dr. Sanford and what I, for example,
it's really not, it's not complicated. And it was unfortunate that like Zach Hancock kept trying to pretend that I meant something completely different by it than what the literature states, because I literally read it right out of the literature. Dr. Walker was very clear that effectively neutral mutations are not affected by natural selection, and yet they can still be deleterious. And that contradicts what Dr. Dan said, and it contradicts, what a lot of people have tried to say tonight. And I guess that's the whole point of what I wanted to do. I wanted to show that if I just show you these dots, these points of data that you can pull straight out of the published literature, the guys that want to deny genetic entropy ultimately have to deny these statements that were made by the established pop gen experts. And that's what we heard tonight. We heard obfuscation and denial of these basic points that they're making.
And these are the quotes that you presented, correct me if I'm wrong, in your presentation, Paul, from Kimura himself.
There's the one from Kimura. Yep.
So is he not saying effectively neutral, but in fact, very slightly deleterious mutants? You're trying to say, okay, he's defining effectively neutral as those being unselectable, but yet still slightly deleterious, not strictly neutral.
Yep. And there's a fitness decline. He tried to put a number on it, 10 to the -7 per generation. That's probably not accurate. That was just an estimate, but the important point is that he agrees there is a decline, and he felt the need to try to appeal to, very rare, mega beneficial mutations that somehow overcome the decline. And that's there's only so many times we can keep going in circles to repeat that. But I hope that at least some members of the audience were edified by what I presented, and that it was helpful for some people. That is my my prayer on that.
Paul, excellent final words. I really appreciate your time. Again, this is 3.5 hours out of your day, out of your time. It also requires a lot of work, a lot of prep. You put together a really fantastic presentation. And so this is very much appreciated by myself, by Sam, by the audience. You're doing great work for the Lord, and I really appreciate it. Sam, any final words, final thoughts from you, my brother?
Yeah, we're just here pointing people to Jesus. So I want to thank you for having me on, and I hope this was encouraging for those of the faith in the chat, and I hope it was challenging for the critics. And I'll agree, there was a lot of I guess semantical disagreement, but the interesting thing about this topic is they're challenging their own camp, because that's who we were citing. So it seems like if there is any confusion, it's within their own camp over there. And not over here. And then the last thing I said, or would like to say, it was nice what Dan's interaction with you. The only thing that I would add is that, you know, it seems like that experiment stopped when they noticed some sort of benefit, as Dan was describing. But if they, as Dr. Sanford rightfully stated, of course, if they continue to let that experiment go, as those mutant in a beneficial way, they would eventually start accumulating those harmful mutations that would eventually lead to their demise also. And that's the argument. Go ahead.
It's what I said all along. We're looking at average absolute fitness over time. We're not looking at maximum fitness over time. That's irrelevant. Eventually that'll go to zero as well. But there are going to be places along that path where you might see some temporary increases in maximum fitness. But what we saw was the average fitness went way down. And if they would have continued that experiment any further, you know, if you get to an average fitness of 0, guess what that means, guys? Extinction. So just because you have some outliers along the way that show an increase of fitness doesn't remove the fact that you have an average overall decrease in fitness. And if you iterate that out indefinitely, you can't have, you know, an average of 0 if you've got any high fitness individuals, right? So eventually that line reaches the x-axis, guys.
Paul, from your engagement with Dr. Zach Hancock, correct me if I'm wrong, it appeared that Zach had a different definition than you or even I on absolute fitness, because I would equate absolute fitness with total functionality. But it did not. There is
a difference there, because he's equating absolute the population genetics literature doesn't care about or look at total functionality.
Right. Yes. Okay.
That's the problem. They only look at reproduction. So the definition of absolute fitness for that, you know, for the literature is going to be only looking at reproduction.
Well, and that's it. I want to point this out. I'm sorry for keeping you longer. I just think this is important. That's why I am thoroughly unimpressed with their broad definition of fitness. And I think you nailed it, Paul. I think you did an excellent job tonight, because a prime example of this is Lenski's experiment. And he measured fitness by testing a single trait, right? Growth rate in a very specific medium. And so they argue that Lenski's bacteria increase in fitness. But when you actually look at what happened, they've decreased in total functional genome size. They've jettisoned just a ton of genes. They've eliminated pre-existing systems. They've really painted themselves into a corner. And so if we look to what you say, absolute fitness or total functionality, we actually observe that they've experienced an overall decline in they're throwing out genes in order to replicate faster, you know? So I completely agree with you that we need to focus on the bigger picture, especially as it pertains to fitness, and the
But-
way population geneticists define fitness in terms of reproduction doesn't really help us in the real world. Isn't that right, Paul?
Well, I mean, it's unhelpful and it's misleading. But at the same time, I want to be clear that even if you are only looking at just reproduction, you know, it's still going to result in extinction. It's just that you're not going to have as good of data. Like I deal with a lot of data, and in my vocation, and I happen to know that, you know, the more data you have to look at to build your picture on, the better. The bigger the picture you've got, the more information you include in your model, the better. But evolutionists have to restrict their model to reproduction only because that's their entire worldview. Their worldview states that the only thing that matters is your progeny, your offspring. That's the only thing that counts. When in reality, we know that that is a vast oversimplification. But even on their model, decline will still happen. It's just that they're not gonna see it as soon because they'll only be looking at reproduction. So there's
Right.
gonna be a time period where, according to their data, things weren't really declining, or they weren't declining very much because they weren't looking at the whole picture. But then by the time they do start to see the decline, it'll be too late. Which is
Because according to the genetic entropy thesis, Paul, eventually enough deleterious mutations will accumulate where the population as a whole has such a high genetic load that reproduction will be negatively affected. Is that right? Eventually, organisms would reach that point.
Of course, absolutely. Yeah,
right.
That's how we get to extinction. But by the time you're so loaded down with deleterious mutations that you can't even reproduce properly, you're way down the line.
Right.
You're messed up, man. You're really
and that's why
messed up at that
selection.
point.
That's why selection's so limited. It involves the death of individuals.
Yeah.
Are you going to wipe away the whole population? That's extinction. That's no help. Or are you just
Isn't it
going to remove a
obvious?
select
Like
people?
there's so many ways you can hurt a functional machine, and so comparatively fewer ways that you can randomly improve on it. It's very obvious, and it's very obvious that you know, there's just no mechanism to get rid of the issues. But anyway, it's getting late, and it's been a good night, but I'm getting pretty tired, so let's call it a night. But I thank you guys for hosting me as always, and I thank everybody who participated tonight.
Paul, God bless. I appreciate you doing this. You've definitely earned the night off and also the weekend off. So you sit back, relax. And this was so fun. We might just have to do another one of these again in the next few months or something. So if anyone's up for it. So, okay, Paul, thank you for doing this. Sam, thank you for doing this. Go share this around. These kinds of conversations are important because it gets us out of our, as I like to say, theological, and in this case, science echo chambers, brings us together with those that we disagree with and engage, especially this topic, very technical, these technical points. So Paul, great job tonight. Sam, thanks for hosting, brother. We'll be back tomorrow for another open mic tomorrow on something theological with the deity of Christ. And to everybody that joined, our critics, we really do appreciate your time and being willing to engage us on this topic. So with that, God bless all. Standing for Truth is out.